Ma'am, please. Dr. Ramu, please, sir. So we start with the first technical session. For this, I would like to request Dr. Ashmi Vidya to chair the session and Dr. Dipti ma'am to please co-chair the session. So please, ma'am and sir, please come up.
A very good morning to all of you. So, I welcome you all in this first technical session. Although our first expert need no introduction, but it's my duty to introduce our speaker with you. Our speaker of the day is Dr. Salesh Sharma, who is working as professor and director in College of Pharmacy, Bela, which is an autonomous college and be accredited UG program. He has 16-year experience. He has done guided 63 M pharmacy students, of which 3 are ongoing, and he's guided 8 PhD students. He has more than 5 patents, 16 copyrights, 109 research papers, and sponsored research project around 1.3 crores from DST, AICT Modro, AICT RPS scheme, and he present more than 15 talks in different conference presentations, and he's a speaker in different platforms. Apart from the award, he is an awardee of Punjab Government Silver and Bronze Certificate, Best Paper Award in Oral and Paper, Best Teacher Award in 2017. Apart from this, is a Young Scientist Award, is a life member of ABTI, IPA, SPUR, IPGA, and is a member Board of Study Pharmacy, IKGPTU. I welcome you, sir. Please, sir. Over to you, sir.
Thank you.
Welcome.
So first of all, this is my duty to welcome you all in the second day of the Spark 2022. So welcome you all in the very podium organized by the Guru Gobind Singh College of Pharmacy. This is my privilege to be a part of this Spark 2022 as a small speaker to deliver my talk on the QBD, the quality by design in pharmaceutical product developments, also in biotechnology. First of all, I am thankful to the management of Guru Gobind Singh College of Pharmacy, Department of Biotechnology, Ministry of India, Dr. Kumar Gaurav and his team to invite me as a speaker to deliver or share my knowledge with you all young, energetic, and my lovable budding pharmacists. So I selected today's topic, the quality by design in pharmaceutical product development. Quality. Quality, it is important in everything. Either it is in education, in food, in dresses. हम किसी भी चीज की बात करें क्या quality important है या नहीं? Yes. Yes, of course, the quality is very important in each and everything. Quality is important for the industry to produce a product. Quality is important for the salesperson to promote the sales of the product. Quality is important for the customer to consume the product. I'm ready to welcome this sir on the second day of Spark 2022. Who's the lucky branch? Or we can So the quality is important by every aspect, by the productions, by the consumer, and by the salesperson. In the next century, a company will stand or fall on its quality and value. Said by the Robert Huss, CEO of Levis, कि वही कंपनी आगे चलेगी जिसके पास क्वालिटी होगी और जिसके पास क्वालिटी नहीं होगी वो कंपनी आगे चल के बंद हो जाएगी. We have very examples in our daily life. Motorola is the first company to introduce the mobile, but today the Samsung and Apple cover all the platforms of the mobile. Why? क्योंकि उन्होंने शायद उस क्वालिटी को मेंटेन नहीं किया जो क्वालिटी वो लेके आए और उसके कारण क्या हुआ धीरे धीरे नोकिया मार्केट से सारे डिसअपियर होते चले. क्वालिटी इज द बेस्ट बिजनेस प्लान. कोई भी आप बिजनेस करते हैं यदि उसके अंदर क्वालिटी है देन यू आर सक्सेसफुल इन योर बिजनेस and then you are fail in your business. Today we all this know about this company. इन सारे लोगों को हम बहुत अच्छी तरह जानते हैं. फार्मास्यूटिकल्स भी है, इलेक्ट्रॉनिक भी है, कंप्यूटर बिजनेस भी है, सर्विसेज बिजनेस भी है, and they are all the qualities motto. इनके आप नीचे देखेंगे इनकी वैल्यू लाइन देन दिस हमारे पास क्वालिटी होगी तो ही हम उसको आगे सक्सेसफुल करें. Importance of product quality for a company, it is because the bad quality product will affect the consumer confidence, product image and sales of the company. एक बार हम कोई सस्ता पेन खरीद लें और उसने भी इंक लिंक कर दी जो हमारी शर्ट पे उसने खराब कर दिया, आप आइंदा से कभी भी उस पेन को नहीं खरीदें. तो इंडस्ट्री के लिए बहुत ज्यादा जरूरी है कि यदि हमारा प्रोडक्ट क्वालिटी वाला होगा तो ही वो आगे सर्वाइव करेगा. हो सकता है एक बार हम गलती कर लें उस प्रोडक्ट परचेज करने के लिए, बट नेवर अगेन यदि प्रोडक्ट की इमेज अच्छी नहीं होगी, सर्विसेज की इमेज अच्छी नहीं होगी तो हम उससे दोबारा डील नहीं करेंगे. सेकंड, फॉर द कंज्यूमर दे आर रेडी टू पे हायर प्राइसेस बट इन रिटर्न दे एक्सपेक्ट द बेस्ट क्वालिटी ऑफ. हमारी इच्छा होती है कि जब पैसा खर्च कर रहे हैं तो हमको अच्छी से अच्छी चीज और सर्विसेज उसके अंदर मिले. So what is the quality product? How can we judge the quality product? बहुत आसान है. हमारे लिए हम कपड़ा खरीदने जाते हैं, हाथ लगा के देखते हैं, कुछ खाने का आइटम खरीद रहे हैं तो छू के देखते हैं, खा के देखते हैं. बहुत सारे हमारे पास ऐसे एंड ऑफ सेंस ऑर्गन्स है जिससे हम पता कर सकते हैं जिसकी क्वालिटी क्या होगी. बट इन द ब्रॉडर सेंस द गुड परफॉर्मेंस ऑफ द प्रोडक्ट, मोर सिक्योरिटी without defect, easy to use and adaptable to specific needs is said to be a good quality product. यदि वो specific criteria को fulfill कर रहा है, these are excellent in this criteria, then we say yes, this product is good for us and it is a quality product. Can we measure the quality? क्या हम किसी भी चीज की क्वालिटी को मेजर कर सकते हैं? Can we check the quality of the mic? आप सबको आवाज अच्छी आ रही है? Means mic की क्वालिटी अच्छी है? आपको विजिबल अच्छा हो रहा है? Then the quality of ये, यदि आपने कपड़े अच्छे पहन रखे हैं तो आपने किसी क्वालिटी के कारण इन कपड़ों को परचेस किया होगा. Means the quality हम चेक कर सकते हैं. Yes. Can we measure the quality of medicine? इंस्ट्रूमेंट, जैसे अभी हमने इनको विदाउट इंस्ट्रूमेंट चेक किया माइक को, स्क्रीन को, क्या मेडिसिन को भी आप चेक कर सकते हैं? मैडम, ड्यूरिंग माय एक्सपीरियंस ऑफ 20 इयर्स आई नाउ चेक द क्वालिटी ऑफ द प्रोडक्ट इन इंस्ट्रूमेंट. केवल हम देख के बता दें कि खराब हो गई या इसके ऊपर ये फंगस आ गई या कुछ और हो गया, बट हम नहीं बता सकते कि जो उस पर लेबल अमाउंट ऑफ द ड्रग है वो उसके अंदर अवेलेबल है या नहीं. We never check the quality of the medicine without any instrument. So it is very difficult to the layman कि वो उसकी क्वालिटी को देख के बता सके कि ये दवाई सही है या no. We cannot measure the quality of the medicine. So who is responsible for the quality of the medicine? जब बाकी सब चीज की क्वालिटी हम चेक कर सकते हैं तो हम उस हिसाब से परचेस कर लेते हैं, बट जब हम मेडिसिन की चेक नहीं कर सकते तो who are responsible for the quality of the medicine? So there are some laws in the universe, in the world, in India, जिनको उसके तहत वो दवाइयां बनानी पड़ेंगी और उन दवाइयों के लिए कुछ नियम, कानून, कायदे बनाए गए हैं जिससे कि जो मेडिसिन बने वो क्वालिटी मेडिसिन बने. In 1960, FDA GMP, 1987 FDA validation, in 2002 FDA CGMP, 2006 ICH, 2008 FDA, again Q8 for product development, 2011 for Q10 pharmaceutical quality system, and many more. जो लास्ट के 5-6 हैं, after the 2006, 2030 up to 2020, these all are based on the quality by design. कि हम जो भी चीज बनाए वो कैसे बनाए with the help of the quality by design. What is the quality by design? Quality by design is a systematic approach to develop a product with the predefined objectives and emphasis on the product understanding and the process controls based on the sound science and quality risk management. To take a very simple example with you, आप सब लोग रोटी खाते हैं, और सबके घर में मदर, सिस्टर, भाभी या आप खुद भी रोटियां पकाते हैं. क्या आपने कभी रोटी बनाने से पहले आटा तोला है? उसमें नमक ऐड करने से पहले नमक को तोला है? उसमें पानी ऐड करते समय पानी को मापा है? उसको प्रोसेस करते समय, भूनते समय आपने क्या उसका टाइम नोट किया है? क्या जब आपने उसको गोल बेला उसका रेडियस चेक किया था, या आपने उसकी थिकनेस चेक करी थी, या जब आपने इस रोटी को सेका था तो टेम्परेचर चेक किया था, तवे का टेम्परेचर क्या है? जब वो पूरी सिक गई तो क्या आपने उसको खा के देखा था कि सही सिक गई है? जब आप जिसको चुपड़ा था तो अमाउंट ऑफ जो घी और बटर जो आप उस पे लगाया था वो आपने नाप तोल के लगाया था? नेवर. अब आप बताइए जो आपने रोटी खाई थी वो क्वालिटी की थी या नहीं थी? ठीक. Why so? कहीं हमने वैलिडेशन यूज नहीं किया, कहीं हमने क्वांटिफाइड नहीं किया, कहीं हमने कुछ भी नहीं किया, पर जो रोटी बनी वो कैसी बनी हमारी? क्वालिटी. एंड व्हाट इज द क्वालिटी सेड? कि इस रोटी को खाने के बाद एनर्जी मिलनी चाहिए, इस रोटी को खाने के बाद शक्ति मिलनी चाहिए. वो उस रोटी ने ये फुलफिल किया. वो रोटी टेस्टी होनी चाहिए, वो रोटी गोल होनी चाहिए, रोटी जली नहीं होनी चाहिए, रोटी स्वादिष्ट होनी चाहिए, नमक बराबर होना चाहिए. These are other parameters. बट पहला पैरामीटर क्या था कि इससे हमें एनर्जी मिलनी चाहिए. डिफरेंस आएगा जब आपकी मदर रोटी बनाएगी, आपकी सिस्टर रोटी बनाएगी, आपकी भाभी रोटी बनाएगी और आप रोटी बनाएगी. You have to use the same आटा, same नमक, same पानी, same बर्तन, same gas, same something, but the taste is varied by hand to hand. उसको दूर करने के लिए वैलिडेशन को लेके आए, जीएमपीस को लेके आए, जिससे कभी भी कोई भी चेंजेस नहीं लगे। और सब में ऐसा कोई है जिसने पार्ले जी बिस्किट नहीं खाया हुआ उसमें? जिन्होंने पार्ले जी बिस्किट खाया प्लीज रेज योर हैंड। पार्ले जी, एवरीवन। आपने कभी देखा है कि वो इंडिया में 17 जगह बन रहा है, 17 फैक्ट्रीज आर इन इंडिया जो प्रोड्यूस कर रही है पार्ले जी बिस्किट। यू फील की है हरियाणा वाला खा रहे हैं, राजस्थान वाला खा रहे हैं, या गुड़गांव का खा रहे हैं, फिर उत्तराखंड का हम पार्ले जी खाने, कभी आपको फील हुआ? वाइस को घर के अंदर मदर से सिस्टर खाना बनाती है तो डिफरेंस आ रहा, वहां डिफरेंस नहीं आ रहा। जबकि इंडस्ट्रीज अलग अलग है, वहां काम करने वाले लोग अलग है, मशीनरी अलग है, रॉ मटेरियल अलग जगह से आ रहा है, बट द फाइनल रिजल्ट ऑफ द टेस्ट विल बी सेम। बस ड्यू टू दी वैलिडेशन। आटा छानेंगे नहीं होगा तो उसको मोटा करेंगे, पानी बराबर मिलाएंगे, गैस बराबर सेकेंगे, बेक बराबर करेंगे, साइज बराबर होगी, तो रिजल्ट जो फाइनल आएगा वो भी सेम आएगा। कंज्यूमर को कभी पता नहीं चलेगा कि वो 17 इंडस्ट्री किस इंडस्ट्री का टेस्ट में उनको कभी भी कोई भी असर नहीं है। सो जब हम किसी भी चीज को बनाना चालू करते हैं और उसके फाइनल रिजल्ट को पहले हम समझ लेते हैं कि जो हमारा रिजल्ट आएगा इस चीज को बनने पर, दिस सिस्टमैटिक अप्रोच इज नोन एज क्वालिटी प्लानिंग। अभी हम क्या करते हैं, द क्वालिटी ऑफ द प्रोडक्ट इज द रिक्वायरमेंट एंड नीड ऑफ द एक्सपेक्टेशन ऑफ द पेशेंट। जो हमें पेशेंट को चाहिए वही चीज हमारे इसमें रिफ्लेक्ट होगी क्वालिटी। देखो होगा मार्केट के अंदर देयर इज अ पाउडर नोन एज इनो, एंड व्हाट इज द टैगलाइन ऑफ द इनो? 6 सेकंड में। पेशेंट की डिमांड क्या थी? व्हाट इज द डिमांड ऑफ द पेशेंट? जब हमारा गैस्ट्रिक हो रहा है, गैस बन जल्दी से खत्म होना चाहिए, ये किसकी प्रॉब्लम थी? पेशेंट की। और कंपनी ने क्या डिलीवर किया? 6 सेकंड। क्या इनो उससे पहले नहीं आ रहा? और तब वो टैगलाइन नहीं थी। जब पेशेंट की नीड बनी फास्ट, तो उसको उन्होंने और जल्दी कर दिया 6 सेकंड। इनो में सेकंड स्टेप आया कि जब लोगों को चाहा कि इसमें फ्लेवर्स आने चाहिए, लेमन, कोला और कई सारे, जीरा, then what they change? They change their quality. उन्होंने अपनी चीज को अमेंड किया और उनको क्वालिटी प्रोडक्ट दिया। आज हम सब लोग भी सिंपल इनो यूज नहीं करते गैस्ट्रिक प्रॉब्लम्स, हम लोग फ्लेवर्ड इनो यूज करते हैं फॉर गैस्ट्रिक प्रॉब्लम्स। सिंपल इनो का यूज करते हैं कहां पर? हम खमण बनाने में, इडली बनाने में, ठीक है। तो उन्होंने उसको सेल पार्ट पे छोड़ दिया और उस चीज को इधर ले गए। ये क्या था? जैसा पेशेंट, जैसा कंज्यूमर को नीड है, उस नीड के हिसाब से हम सामान बनाएंगे तो हमारा क्वालिटी प्रोडक्ट होगा, एल्स हमारा क्वालिटी प्रोडक्ट वो नहीं होगा। Quality by design to ensure the high level of assurance of product quality for the patient, more efficient by the regulatory oversight। यदि हम किसी भी चीज का डिजाइन क्यूबीटी के थ्रू बनाते हैं तो उसको समझाना बहुत आसान है, उसका एक इनसाइट लेवल को देख पाना बहुत आसान है। जो हमारी रेगुलेटरी अथॉरिटीज है जिनको अप्रूवल देना है, उस चीज को बहुत अच्छे से और जल्दी समझ सकती है। देन एफिशिएंसी एंड कॉस्ट सेविंग फॉर द इंडस्ट्री। The aim of quality by design क्या आप यूज कर सकते हैं? This is active pharmaceutical ingredient industries, material and excipient industries, analytics, simple dosage form designs, advanced drug delivery systems, developed devices, combination products, and biotechnological products. Now, what is the difference between the traditional and QBD? In the traditional, when we start the development of any product, तो पहले हम उसको बनाने का कोशिश करते हैं, उसके बाद उसको इवेल्युएट करना स्टार्ट करते हैं। पाउडर मिक्स किया, देन वी हैव टू चेक मिक्सिंग हो गई कि नहीं, उसको गरम किया, देन वी चेक ये गरम हो गया कि नहीं हुआ। After every step we have to evaluate it, but in the QBD there is no need to evaluate after these steps। केवल आप फाइनल प्रोडक्ट को चेक करेंगे, जो आपको चाहिए था वो उसके अंदर मिलेगा। The current approach quality is assured by the testing and inspection। हर बार हम उसे टेस्टिंग करनी पड़ती है, उसका इंस्पेक्शन करना पड़ता है। This approach is known as the current approach जो we use कर रहे हैं। QBD approach is build the quality to the product by decision based scientific understandings। It is data intensive submission। जब हम इससे कोई भी चीज का रिजल्ट निकालते हैं तो उसके लिए बहुत बड़ा डाटा लग जाता है, जबकि यूबीडी के थ्रू बनाते हैं तो छोटे से डाटा को बार बार यूज करके अलग अलग तरह से चीज को यूज करते हैं। स्पेसिफिकेशन बेस्ड ऑन बैच हिस्ट्री, जो पुरानी बैच हिस्ट्रीज है उनको बार बार यूज करके हम करंट अफेयर्स में यूज करते हैं, जबकि स्पेसिफिकेशन बेस्ड ऑन प्रोडक्ट परफॉर्मेंस, कि हमें प्रोडक्ट का फाइनल रिजल्ट क्या चाहिए उसको हम चेंज करके यहाँ पर दियर को यूबीडी से चेंज कर सकते हैं। फोकस ऑन द रिप्रोड्यूसिबिलिटी, हमेशा एक ही डर रहता है कि वही रिजल्ट आना चाहिए जो पहले बैच में आया था। हम लोग यूज करते हैं इस टर्म को बेस्ट टू बेस्ट कंसिस्टेंसी, जिसको हम बनाने की कोशिश करते हैं इन द करंट अप्रोच, बट फोकस ऑन द रोबस्टनेस, अंडरस्टैंडिंग एंड कंट्रोलिंग द वेरिएशंस इन द रिजल्ट्स, उसको हम यूज करते हैं इन द यूबीडी। जब भी हम किसी चीज को बनाते हैं, जैसे हम लोग बात कर रहे थे इनो का टैगलाइन था 6 सेकंड, ऐसे इनो ने अपनी टैबलेट मार्केट में लॉन्च कर दी इन्होंने इनो टैब, वो आपको कह रही है कि विद इन 30 सेकंड्स बिल्कुल गैस्ट्रिक प्रॉब्लम्स को दूर कर देगी। तो जो टैबलेट 30 सेकंड के अंदर टूटनी चाहिए तो उसका मेन एंड क्या होगा, व्हाट इज द टारगेट प्रोडक्ट प्रोफाइल, वो टैबलेट कैसी होगी जो 30 सेकंड के अंदर डिसइंटीग्रेट आउट हो जाएगी, डिसॉल्व हो जाएगी। तो हमारा टारगेट प्रोफाइल क्या हो गया, द टैबलेट व्हिच में डिसइंटीग्रेट इन 30 सेकंड, दिस इज टारगेट प्रोफाइल। जो भी हमें फाइनल रिजल्ट उस चीज का चाहिए डोजेस फॉर्म का, वो उसका क्या कहलाएगा, टारगेट प्रोडक्ट प्रोफाइल। फॉर एग्जांपल एनी फार्मास्यूटिकल टैबलेट में डिसइंटीग्रेट विद इन 50 सेकंड, द प्रोबेबिलिटी इज लेस देन 0.6 एंड डिसोल्यूशन मोर देन 95 मिनट्स। These are the final results for the इनो टैब। जो इनो टैब है उसका फाइनल रिजल्ट था, ये हमें रिजल्ट चाहिए जो हमारी टैबलेट बन गया है, then these are known as target product profile, की बात we have to target it, कि हमारा फाइनल रिजल्ट क्या होगा। These are the simple control any tablet, appearance, size, shape, weight, hardness, stability, disintegration, assay constraint, resolution, pharmacokinetic studies and their stability. These are some specific features understand करने पड़ेंगे, इनको यूज करना पड़ेगा। These are the stability, disintegration and dissolution and other are known as the non-specific parameters। यदि उसमें कोई changes होता है no issue, but जो हमारा टारगेट प्रोफाइल है वो सेम रहना चाहिए जो उसको लेके चले। These are the various inputs, the raw material, people, equipment, process, materials, environments, जिसकी बार-बार हमको वेरिएशंस मिलेगी, but we have the output will be the same। हमेशा हमें सेम चाहिए, उसके लिए क्या करना होगा, we have to जो हमारे प्रोसेस पैरामीटर्स हैं और जो क्वालिटी एट्रीब्यूट्स हैं उनको यदि हम मैच कर दें आपस में, then they gave the similar results and the desirable results at the time when we have to get those outputs। So किसी भी हम टैबलेट पे भी बना रहे हैं तो उसके अंदर DT disintegration time is responsible by the disintegrant 1 and disintegrant 2। यदि हम प्रोसेस में देखें then this is the tableting, जब उसके हम पंचिंग कर रहे हैं तो वो एक ऐसा प्रोसेस उसके disintegration टाइम सेट करेंगे। Then the friability by the disintegrant 1 and disintegrant 2, and again the tableting process is responsible for the friability of the tablet। The dissolution is based on the APR, disintegrants and tableting process, कि उसमें से कितनी चीज बाहर निकलेगी, कितनी चीज उसमें से सॉल्व होगी, डिसॉल्व होगी। These are the three parameters जिसके अंदर API है, disintegrant 1 है और disintegrant 2 है, which is responsible for the DT, friability and dissolution। Similarly, the tableting is the process parameters जो हमारी टैबलेट की disintegration, friability और dissolution को affect। कुछ materials हैं जो affect कर रहे हैं, कुछ process है जो affect कर रहे हैं। यदि हम इन process और materials की study अच्छे से कर लें, then we decide कि हमारा DT क्या होगा, हमारा friability क्या होगा, हमारा dissolution क्या होगा, means कि हमारा जो TPP है, target product profile, वो अंततः क्या आएगा, last क्या होगा। Design space, ऐसी हो कि यदि हम किसी भी डिसइंटीग्रेंट की बात कर रहे हैं तो उस डिसइंटीग्रेंट का एक अमाउंट हमारे पास लिटरेचर में ऑलरेडी गिवन है कि 1% से लेके 10% तक यदि हम इसको यूज करते हैं तो टैबलेट विद इन 30 सेकंड 1 मिनट के अंदर डिसॉल्व आउट हो रही है, डिसइंटीग्रेट हो रही है। एंड दिस इज नोन एज द नॉलेज स्पेस। एक बड़ा है, दिस इज नोन एज नॉलेज स्पेस, जो हमारे पास बुक्स में लिखा हुआ है, फार्माकोपियस में लिखा हुआ है, एक्सीडेंट बुक्स में लिखा हुआ है, पुराने कई सारे लिटरेचर्स के अंदर मौजूद है। Then this is our design space. 0 से 10% के बीच में हम कितना यूज करना चाहिए? It is 1% to 6% area we say use, then this is known as the design space. Then the control space कि हम कितना हिस्सा इसका यूज करें जिससे कि मैक्सिमम रिजल्ट जो है हमको वो मिल जाए, then this is known as control space. यदि उसके बाहर की कोई नई हम खोज कर लेते हैं, then this is known as the explore space. यदि हम इसको 5% तक था, यदि हम इसको 8% डाल दिया तो it will use as a binder also. तो ये हमारे लिए एक्सप्लोर स्पेस बन जाएगी इसमें. दिस इज अवर सिंपल फॉर्मुलेशन टेबल जो आप लोगों ने भी फार्मास्यूटिक्स में पढ़ी होगी, बनाई होगी, जिसके अंदर दो चीज को हमने वेरिएबल किया एट ए टाइम. डिसइंटीग्रेंट 1 को 2 4 6 8 डाला. देन सेकंड वाली टेबल में फॉर्मुलेशन 5 to 9 के अंदर 5 10 15 20 हमने इसके अंदर मिक्स किया. और लास्ट की कुछ फॉर्मुलेशन है जिसके अंदर हमने डिसइंटीग्रेंट 1 और डिसइंटीग्रेंट 2 का दोनों का कॉम्बिनेशन यूज किया. Now what will happen with that? यदि हम इसका रिजल्ट देखें तो हमने जो TPPs ली थी, उन TPPs के अंदर the disintegration time is 124 to 45 by using the one disintegrate, and in second it is 172 to 56, and in the both combination it is 101 to 41. but जो हमारे डिमांड थी 50 सेकंड, वो इनमें से किसी में भी नहीं आ. ये जो हमें टारगेट प्रोफाइल चाहिए थी, जो हमारी टैबलेट थी 50 सेकंड के अंदर डिसइंटीग्रेट हो, वो हमको कहेगी नहीं मिल रही. There is no results for 50 seconds of the DT and probability is 0.6. So what will we do? हमने अभी तक 12 फॉर्मुलेशन बना ली है, पर जो हमें रिजल्ट डिजायर्ड था वो हमें नहीं मिल रहा. तो हम क्या करें? We increase the points, हम इसको बढ़ाते जाएं. और अभी हमने 13 बनाए, अब हम 20 बनाए. और means we change the one factor and got to the result. अगर एक को चेंज करें, रिजल्ट तक चेंज करें, रिजल्ट तक पहुंचे, we can say we will use the hit and trial method. हिट करते रहो, जहां पर अटक जाए then good, नहीं है तो फिर चल ही रहा. तो हिट एंड ट्रायल मेथड का यूज यदि हम करना चाहते हैं, this is known as one factor at a time. If we make some graph कि जैसे जैसे हमने डिसइंटीग्रेंट का अमाउंट बढ़ाया तो स्टेबिलिटी क्या, डिसइंटीग्रेशन पे क्या फर्क पड़ा. यदि इसको हम कंबाइन कर दें सारे, रिकॉर्ड में लगभग कि हमें एक ऐसा पॉइंट मिलेगा जहां पे चारों लाइन मिक्स कर रही है. इट नॉट गिव्स द 20.6 and this is the point जहां हमारा 70.6. तो what will we do? जो हमने रिजल्ट बनाए उससे हमको वो अचीव नहीं हो रहा जो हमें चाहिए था, कंपनी को वो चीज अचीव नहीं हो पा रही जो उनको चाहिए थी. Then what will we do? We use the design, we use the quality guidance. What is this? This is a holistic mathematical system-based approach to design the development and delivery of the best product under the given of set solutions. These are some software-based programs where we use that and produce a desired product जो हमको चाहिए. If we have two factors, then this is response surface. If we have three factors, then central composite design, box Wilson design, box packet design. Now what they do? When we prepare this formulations, यदि हमने यहाँ पर दो formulations ली हैं, दो हमारे variables लिए हैं. The first variable is disintegrant 1 and second variable is disintegrant 2. उसके तीन levels हमने डाले. -1 is low, 0 is medium and +1 is the high. इसके हम combinations बनाए, then हमारे पास 3 levels हैं, 2 factors हैं, तो 3 का square 9. We have to prepare only the 9 formulations. वो 9 formulations हमने बना के उनका details probability lab में निकाला. क्या होगा? Simply जब हम डाटा को सॉफ्टवेयर के अंदर अपडेट करते हैं, then they will give equation for that. Equation means it gives the mathematical model for the system. आज से पहले घर के अंदर जो टेप रिकॉर्डर्स हुआ करते थे या घर में जो पुराने टीवी हुआ करते थे, तो उनकी वॉल्यूम जब हम बढ़ाते थे तो वहां पर रेगुलेटर सा बना रहता था, उसके ऊपर बढ़ता जाता था, पर हमें पता नहीं था कि कितनी वॉल्यूम बढ़ गई। जब मोबाइल्स के अंदर एलेक्सा के अंदर एलईडीज के अंदर देखते हैं डिजिटल, क्या आपने 52 लेवल कर लिया है अपना वॉल्यूम, 56 कर दो, 58 कर दो, 82 कर दो, 80 इस आपने उस चीज को क्या कर दिया, सारा का सारा डिजिटलाइज। एंड नाउ व्हाट इज द इफेक्ट ऑफ D1 एंड D2 ऑन DT? वो क्या आ गया यहाँ पर हमारे पास, ऑन द बेसिस ऑफ डिजिट्स। तो जो चीज थी वो क्या इफेक्ट दे रही है उस टैबलेट के अंदर, वो हमने इससे विद द हेल्प ऑफ सम सॉफ्टवेयर उसको हमने कन्वर्ट कर लिया विद द इन द फॉर्म ऑफ डिजिट। कि डिसइंटीग्रेंट 1 9 टाइम्स DT को घटाएगा एंड डिसइंटीग्रेंट 2 decrease the DT by 22 times. Means disintegrant 2 is more powerful to break that tablet than disintegrant 1. ये हमें कहां से मिल गया, with the help of the mathematical modeling. And simple. यहाँ पर यदि हम DT के वैल्यू डालते हैं 50, then we can identify कि at the value of 50 हमें D1 और D2 कितना डालना पड़ेगा. So I welcome the team members of Spark 22, Dr. Bharat Parashar, doctors from Jaipur, Rajasthan. Similarly for the release, similar for friability, the D1 effect is 0.08 on the friability and 0.33 is the effect of D2 on the friability. We mix it. जो हमने पहले ग्राफ बनाया था उसके अंदर ये पता कि जहां पर टैबलेट का डिसइंटीग्रेशन 50 होगा और फ्राइबिलिटी 0.6% होगी, देन वो पॉइंट हमको नहीं मिल पा रहा था विद दिस सिंपल ऑफेट डिजाइन जो हम हिट एंड ट्रायल मेथड यूज कर रहे थे, वहां तक हम नहीं पहुंचे। बाय यूजिंग द 9 फॉर्मुलेशन वी रीच एट द पॉइंट वेयर वी गॉट द x1 की जो हमें वैल्यू डालनी है डिसइंटीग्रेंट 1 की, इट इज 0.77 and disintegrant 2 जो हमने डालना है it is 0.74. Then we got the desired results 50 second and 0.60% of the friability जो हमें ऑफेट में नहीं मिल रहा था। With the help of QBD we reach up to the point और वो हमें मिल सकता। Now what is the major benefit of this thing? आज हमारी कंपनी ये टैबलेट मार्केट में उतार रही है जिसका DT 50 सेकंड है और जिसकी फ्राइबिलिटी 0.6 है। The next day, our competitor says our tablet will be dissolved in 30 seconds. Then we don't to prepare again batch, start the batching. Similarly, we change the values DT 30 seconds and we reach to the 30 second. और वहां पर हमें नई क्वांटिटी मिल जाएगी कि अब आप D1 कितना डालो, D2 कितना डालो, तो हम उसको उसी टाइम कॉम्पीट आउट कर सकते। So this is the benefit of the QBD कि बार-बार हमको बेसिक और जीरो से स्टार्ट नहीं होना पड़ेगा। We reach some platform to change our quality product according to the need of the patients or the consumers. So these are the 3 possible solutions given by this software जिसके अंदर DT 50 है 0.6 है। यहाँ पर जो हमें मटेरियल सस्ता लगेगा क्योंकि हमें यदि मार्केट को उतारना है तो उसके लिए भी होना चाहिए कि वो चीज सस्ती भी हो, चीप भी हो, अफोर्डेबल भी हो। Then जो चीज इसमें महंगी हो रही है D1 और D2 उसको हम छोड़ सकते हैं, दूसरी वाली चीज को ज्यादा अमाउंट बढ़ा सकते हैं, पहली वाली को कम अमाउंट पे कर सकते हैं। Then design of experiment यदि हम इसे कंक्लूड कर दें then it is a useful screening of variables with significant impact, classical approach then effect। हमने वहां पर अभी तक 16 फॉर्मुलेशन बना ली थी तब भी हम रिजल्ट तक नहीं पहुंचे थे, यहाँ 9 फॉर्मुलेशन बना के भी हम रिजल्ट तक पहुंच गए थे। That it is a limited number of the experiment with the huge information. DOE helps to study the effect of interaction of multiple factors at a time. Used in the optimization studies enable creation of target space. Target space is proposed by the applicant and subject to regulatory assessment and the approvals. Target space developed at the lab or pilot scale can be proposed for commercial scale but need to be verified at the production scale for scale dependent parameters also. So it is our minimal approach, it is reactive, problem solving and corrective action, but in the QBD approach, it is a preventive action. And there is a continuation improvement facilitated. There are 2 ways to reach from one desert to the another one and one mountain to the another mountain. बीच में खाई है, पानी है, समुद्र है, यदि हमें वहां तक रीच करना है तो हमारे पास दो रास्ते होंगे. One is the planned way, second one is the unplanned way. Unplanned way, we jump into the samudra and reach to the second mountain. कब पहुंचेंगे, कैसे पहुंचेंगे, कितना टाइम लगेगा, कितनी मेहनत लगेगी, कोई आइडिया नहीं. एंड सेकंड वे, यदि हम इस पे ब्रिज बना लें, देन वी हैव टू से 1 किलोमीटर ही हमें चलना है, कितनी स्पीड से जाएंगे, विद इन 5 मिनट्स हम वहां पर रीच कर जाएंगे. देन दिस इज द डिफरेंस बिटवीन द ऑफर टेक्नोलॉजी जो आज हमारी इंडस्ट्रीज यूज कर रही है, एंड सेकंड वन इज द क्यूबीडी टेक्नोलॉजी जिसके अंदर हर चीज हमारी आइडेंटिफाइड है. तो व्हाट इज द इंपैक्ट ऑफ द क्यूबीडी? क्यूबीडी का इंपैक्ट क्या है? इट इज academics change their curriculum. PCI ने सारा अपना सिलेबस कॉमर्स का चेंज कर दिया है और उसको पूरा का पूरा क्यूबीडी के ऊपर लेके आ गए. बच्चों को अब क्यूबीडीज पढ़ाना शुरू कर दिया है. इवन 8th सेमेस्टर बीफॉर्म के अंदर भी सब्जेक्ट के अंदर इंट्रोड्यूस कर दिया है क्यूबीडी, क्योंकि दिस इज द डिमांड ऑफ द टाइम, डिमांड ऑफ द यार ये आना ही चाहिए. सेकंड, रिसर्च न्यू मीनिंगफुल सॉल्यूशंस आर गिवन बाय द क्यूबीडी. Industry change their research methodology. Industry ने बिल्कुल change कर दिया, offer technology बिल्कुल use नहीं कर रहे, they use only QBD technology. Regulators also change their laws. पहले वो कहते थे कि बड़ा डाटा लेके आइए तब हम देखेंगे, आज वो कहते हैं कि यदि आप छोटा सा QBD से यदि कोई डाटा लेके आए तो हम उसको भी मान्यता उतनी देंगे जितना आप पहले लेंगे. So these are the usefulness or impact of the QBD on different areas of the pharmaceutical research. So regulation is quality thinking. Quality cannot be tested in the product. हमें किसी चीज को देखने की जरूरत ना पड़े कि क्वालिटी होगी या नहीं. It has to be built by design. उसको हमें डिजाइन के जरिए उसके अंदर डालना ही पड़ेगा. आपके पास दो वाटर बोतल्स हैं, वाटर बोतल का डिजाइन सेम है, दोनों पैक्ड है, बट एक के ऊपर बिस्लरी का लेबल लगा हुआ है, एक पे बिस्लरी का लेबल. कौन सा पानी पिएंगे? इस तरह की बोतल थी, एक का हटा दिया, मैं कह रहा हूँ, तो आप कौन सा पिएंगे? लेबल वाला. वाह, आपको मेरे से ज्यादा भरोसा था बिस्लरी के. उन्होंने अपने ब्रांड की वैल्यू बनाई बाय द क्वालिटी. आपने अपने खास आदमी का भी विश्वास नहीं किया कि ये पानी बिस्लरी का होगा, जबकि मैं कह रहा हूँ कि मैंने उस पे से लेबल हटाया है. दोनों बोतल सेम थी, एक पे से लेबल हटा दिया और एक लगा हुआ है. आप कौन सा बोतल यूज करेंगे पीने के लिए? यू से जिसके ऊपर बिस्लरी का लोगो लेबल लगा हुआ है उसी को यूज करेंगे. आपके कहने से भी हम उस बोतल को यूज नहीं करेंगे जो बिल्कुल क्वालिटी के बराबर. सो क्वालिटी कैन नॉट बी टेस्टेड इनटू द प्रोडक्ट. उसके नाम से लिखनी चाहिए कि यस इट इज द क्वालिटी प्रोडक्ट. सेकंड, इट हैज टू बी बिल्ट बाय द डिजाइन. So thank you very much for your patience listening. Yeah, my email ID is salish.bela@gmail.com. A contact number with Dr. Ashwini and the team of DGSCOP. यदि आप कभी भी इसमें कोई सॉफ्टवेयर पर रिसर्च कर रही हो या अपना प्रोजेक्ट कंप्लीट करना हो इस चीज को लेके, always our gates are open for all of you. Thank you very much.
So first of all, a very good morning to all the dignitaries on the dais and off the dais and all the students who are present here. So Dr. Salesh has explained quality by design with the examples of household codes example that we heard. So he has wonderfully explained what is quality, what is validation, and what we mean by QBD. So if you have absorbed half of the lecture also, that would be wonderful for all the students who are present here. I just have one question with Dr. Salesh. Like from 2013 onwards, the QBD, like you have said, we expect that QBD should be in the generic drug manufacturing. So what is the status in India?
Madam, in the India, mostly all the FNDs, we can say FNDs, nowadays use the QBD technologies for the formulation of the tablets, capsules, or all the pharmaceutical products. They have used the target product profile, what they want for the specific country, for the specific needs of the country. They change their formulation by using the QBD and produce the medicines for each and every.
So actually in India we have a big
generic business here, and we have small
industry
manufacturers. Yeah.
Yeah.
So is it challenging for them?
No,
Like how it is the
all the generics are prepared by the third parties manufacturers, but the formula is given by the big companies which have their own R&D and appendix. First, they set some pilot plant scale, then they get scale up in the company of the third manufacturing space, and then given the formula to produce the tablets. जो हमारे यहाँ जेनरिक बन रही है उनको पूरा एक फॉर्मूला सेट करके दिया जा रहा है उनकी इंडस्ट्री के हिसाब से, जिससे कि उनकी जो टारगेट प्रोडक्ट प्रोफाइल है वो उनको पूरी मैच होती है.
Thank you so much. Thank you. All the students here should take this message. अगर आप लोग entrepreneurs भी बनना चाहते हैं, तो how you can take out good points from this lecture. Thank you so much. Thank you.
If anyone has any questions, you may also ask. So now I would like to request Dr. Rashmi Ingran ma'am, please felicitate sir. So I request you to welcome some of our resource persons who have just arrived. Dr. Abhishek Tiwari, Professor, IITM University, please sir. Dr. Varsha Tiwari, Professor, IITM, please ma'am. Ritika Kera, Assistant Professor, Ambani College of Engineering. Thank you very much, sir. Thank you, Varsha ma'am. So for the next technical session, I would like to invite Mr. Abhishek Tiwari sir and Mr. Dr. Rahul. To Dr. Abhishek Tiwari sir and Dr. Rahul to please chair and co-chair the session. Please, sir.
Morning to all the delegates, faculty members, and dear students. Today we have Dr. Vinay Gathar sir, Professor, MIT Institute of Pharmacy, MIT University, Noida. He is giving his talk on artificial intelligence and drug discovery and development. Sir is having a total experience of 21 years of industrial academic research experience in pharma sciences, drug discovery and development. Sir has guided 6 PhD students, 50 farm scholars, and currently supervising 6 PhD scholars. His key areas are medicinal chemistry, molecular docking, bioinformatics, nanoparticulate drug delivery, expertise in his expertise in drug delivery systems like enantiomer, mesoporous silica nanoparticles, solid lipid nanoparticles, self nanoemulsifying systems, analytical method development. The research expertise of Sir is development of nano platforms of drug delivery, analytical tool, extensive experience with in silico tools like structure-based drug design, talking pharmacophore modeling, QSR, molecular dynamics, homology modeling, drug delivery, and development of therapeutic target like cancers, various CDKs, CHKs, GSKs, MMPs, inflammation, PDEs, COX, diabetes and metabolic disorders like GK, GSK, and PAPs, Alzheimer's, antimicrobial resistance. Sir is having more than 125 national and international publications with Scholar H-Index, Google Scholar H-Index of 25 citations, 2,938, I-Index 55, Scopus Index 22, and cumulative factor of 3 entries. Sir has published more than 10 book chapters in international publishers. Sir received a such grant of 19 lakhs from RPS scheme in 2013 to 16, worked as OPI from 2012 to 14 with and received 15 lakhs of grant, completed postdoc fellowship from FCT Portugal, Best Scientist Award from Jubilant Biosys Limited in 2006, selected for Young Scientist Scheme 2000 to 2006 by Department of Science and Technology, Delhi, gold medalist MPharm 2002, GATE qualified in 2000 with 88.18 percentile, received a travel grant from DST, and his current research areas includes nanotech delivery for therapeutic areas like cancer, autoimmune disorders, AMR, drug discovery and development for autoimmune disorders, AMR and cancer, assessment of pharmaceutical residues and environmental resources. So I heartily welcome Sir for his much time.
So, first of all, thank you, Dr. Devaki and Dr. Sponsered. Good morning, all of you. I would like to thank the organizers, Dr. Kumar Gaurav and his team, Dr. Ashwini, Dr. Geeta, Dr. Ajmer, for inviting me on this talk, for giving delivering a lecture on artificial intelligence. How many of you are familiar with the artificial intelligence? I think nowadays everyone is carrying a device which is artificially intelligent. You just say, "Hey Siri, hi Google, can you take me to this? Can you take me to that? Can you call this fellow? Can you call this?" So we all have that device with all of us, which is based on one of the last revolutionary technology of 10 years, I would say, that is artificial intelligence. So what exactly is artificial intelligence? It is nothing, it's a kind of a computer. We are making that, we are commanding him, we are giving him instructions, we are making him to analyze human brain and then deliver the output. That is what exactly is artificial intelligence. It's not like something which is coming from space or anything is happening. No. It is basically it's a computer. We are using that, we are making him to perform the functions which will identify our brain, which will sense our brain, and that's what we are doing in artificial intelligence. And now, how we can use that artificial intelligence in business? That is another challenge. So first of all, first of all, let's understand what is the need of drug discovery. Why there is a need of drug discovery? The research and development companies, even the research and development academic institutes, they are spending so much of money in discovering new drug molecules. So why they are spending so much of money? Ultimately, they are trying to find out. They are trying to find out the solution for the diseases. They may be the existing diseases. They may be the new diseases. You have seen recently there was a major disease which came into existence last 2 and 3 years. We all suffered and still suffering. Probably, I think every student or every person who is sitting in this auditorium, they or some of them, their family members, they have suffered with that.
Bishop, sir?
They face that challenge in one way or another. All of you have faced that. Maybe the mild symptoms, maybe the major symptoms. Many things happen. So that was the new disease which came into existence. Of course, it was not completely new, but that was there. And then we are facing lots of diseases which are prevalent from so many years. You can imagine like diabetes. We have the problems with central nervous system disorders. We have the major problem, the cancer. There is another disease, simply I have represented here. That is the autoimmune diseases. I think with this disease also everybody is suffering. More or less every human being, they are suffering with this disease also. In one way or another. Maybe it may be the diabetes, maybe the arthritis, maybe these all are there. Systematic lupus, rheumatoid arthritis, multiple sclerosis. So all these are the diseases which are caused by the autoimmune system. Our immune system is changing. Okay, so all that, while this is one major challenge here. Apart from that, there is another major issue which is nowadays very common. Last 10 years, we have seen another term which is known as AMR. Nowadays, there is a on FM, if you will go, they are completely going through the advertisement kind. Or they are making sensitized that be careful about antimicrobial usage. Otherwise, you may face the challenge of antimicrobial resistance. Cancer is another thing. Then there are another so many different viruses, bacteria, parasites, and all those things are there which are causing the diseases. So if we look into the diseases, we can say basically we have two types. One is extrinsic factors, which are causing the diseases. All your different microorganisms and all they are causing the diseases. So they are extrinsic. And we have intrinsic, where our body is changing, our metabolism is changing, our genes are being influenced by the exposure to different things. For example, your lifestyle. Okay, the lifestyle is making us to change. Our genes they are changing. Hormonal influences, environmental factors. Okay, pollution. Everybody is facing this challenge also. Pollution, everybody like we have it is being said now that the kind of weather we are facing in the during the winters, beginning of the winters, we all know that is how much infectious it is, how much it can damage our lungs. And we can have lots of lung diseases, asthma and all, and it is very dangerous for us. So that is there. So that's why these all the diseases are, and for them we need to develop the new molecules. We need to develop the new drug molecules. We are changing the properties of the molecules also. For that also we need to develop. Suppose a drug is not important. I want to make a potent drug. A drug with 5 mg can produce the similar. Why I will need 500 mg? So definitely I will prefer the 5 mg. It will be having less results maybe. That's why I want to develop a new drug. These all are the challenges that we need to develop. But what is there in those that disease? What exactly is happening in those diseases, which are intrinsic or maybe the viruses or all. Ultimately, that is the genetics. So first of all, if we can understand our genes, probably we can develop the drug molecules by targeting the products which are being formed by the genes. That's why there is this term, one is genomics. This genomics. If we can understand the entire genetic sequence of us or maybe any other organism, so that is genomics. Okay, human genome. It was resolved in 2003. There was a long research that it took approximately 13 years to identify the genetic sequence of our DNA. So our DNA is made up of 3.2 billion base pairs of nucleotides. 3.2 billion. And that contains approximately 25,000 genes are there, which are basically responsible for generating different types of proteins, enzymes, receptors in your all different receptors, which are protein in nature. So most of you might have studied this thing, that what are receptors, how a drug acts, how it produces its mechanism of action. It produces its mechanism of action by binding with different types of receptors. What are those receptors? From where they are originating? Those receptors, they are basically originating from our genes. These genes are our genes. I told you that 3.2 billion base pairs of nucleotides. Out of that, 25,000 genes are there, which undergo the process of transcription, and they generate different type of mRNAs. And those mRNAs, then they generate the different types of proteins, that is the translation process. And those proteins, they are the machineries of your body. They function in your body, they regulate different metabolic processes. So these processes, these are like from your food, you generate glucose. Okay, we are not taking directly the glucose, it is being decomposed, it is being metabolized. Carbohydrates are metabolized, they are converted into glucose. Similarly, your fatty acids, we are not taking directly the fatty acids. We are taking the long chain fatty acids, they are being broken down into smaller ones, and then they are used for the different purposes. So they are built up to developments, the layer of the fats. So that is metabolomics. What are the products which are generated? What are the different chemicals which are there in our body which are functioning? Okay, your brain is passing the signals to your all the functions you are carrying out. How the brain is performing. So brain is sending some signals. Signals are in the form of the chemicals. Signals are in the form of some hormones. Signals are in the form of some peptides. Signals are in the form of some proteins. That is, the signals are coming, and then we are analyzing those signals. Now, bring those signals back to the brain analyzes, and then we interpret. This is the mechanism exactly we understand. So, metabolomics, that is the study of what are the different types of chemicals, what are the different types of hormones, all which are the products of metabolism by different types. So we have to understand that. If we can understand that, maybe we can do more development knowledge. So as I told you, human genome 25,000. If you look at the mouse, 24,000, almost very close to human beings. Why we carry out the experiments on small animals? Okay. In pharmacology, you all are being taught that do this experiment on small animals. Why? So there is some similarity with our genetic system and the in humans and the mouse, the rats and mice which we're using. We have the similar genetic, similar types of genes, similar type of enzymes which are there in our body, probably they are present in ours. So that's why we use them as the animal models. Similarly for others, so if suppose you have to target a virus, you have to understand that. Suppose you have to treat a microorganism, you want to kill it, you want to make it inactive. So you need to understand the genetic system of the microorganism. If you need to, if you have to kill mosquitoes, so you have to understand the mechanism. What is there in mosquito that I can target, and the drug will act on that system and it will inhibit. It will kill the mosquitoes. This exactly we are doing. So what are drugs? Because we are using for the treatment of different diseases. Drugs are basically, you can say there are two types of molecules. One is the agonist, there is of course partial antagonist and partial agonist, inverse agonist, all those terms are there. But we have to majorly do two things, agonist and antagonist. One thing is agonist, they will bind with the receptor, they will activate it, and it will produce its mechanism of action. Another one is antagonist, that will bind with the receptor, it will block the receptor, and it will stop it to perform that function. So this is the difference between agonist and antagonist. There are four major classes of receptors. One is the protein coupled receptors. Second is the ligand ion channel receptors. Another one is enzyme linked receptors. And the last one is the nuclear receptors, or we call it as steroidal receptors. Why? So these four are the major categories of our receptors, and they are being produced by the receptors are what? Receptors are basically proteins. All your receptors are proteins. So if we understand, that's why if we can understand what is the protein sequence, amino acid sequence of these receptors. So that study is known as proteomics. Our body is producing more than 100,000 proteins. 100,000 proteins, they are functioning as different, they are carrying out different functions in our body. They are carrying out different functions. Okay. Ligand ion channel receptor, antihypertensive drugs, how they produce their mechanism of action. They produce their mechanism of action by binding with beta adrenergic receptors, okay, which are present on our blood vessels. So they block that. They act as the antagonist, the beta adrenergic receptors. They will block it. And because of that, adrenaline will not bind with those receptors and it will not increase your blood pressure. This is the mechanism of it. So now, if you have to design a drug which has to act on the beta adrenergic receptor, what you will do? Probably, first of all, you will understand how adrenaline is binding with the beta adrenergic. If we can understand that thing, probably it's very easy to develop a new drug for beta as if beta adrenergic. This is exactly we have understood. So what is the goal of drug discovery? If you look at the number of chemicals which are available, one more thing I want to mention is that drug molecules, basically nowadays there are two types of drug molecules. One of them is known as the small molecules. Okay, small molecules which are having their molecular weights less than 1000. They are the small molecules. But last 15 years, last 10 to 15 years, there is a complete change. We are observing that lots of biotechnology derived products are coming into market. They are being used in the treatment of cancer. They are being used in the treatment of arthritis and other problems. Biotechnology derived products, which are basically proteins in nature. They are antibodies which are developed in our systems. We use them as the drugs. Insulin. Everybody is familiar with insulin. Insulin is what? Insulin is used as a drug. It's a drug only. Insulin is what? It's a peptide. It is made up of 21 amino acids. It is a drug, but it is a peptide drug. Why we are not able to develop the system oral where we can take insulin by oral route? That is the reason that insulin, whenever you will take it by oral route, it will be immediately metabolized into your gastric system, and it will not be absorbed into the blood, and it will not reach to its site of action. That's why it is being injected, and then it binds with its receptor state. So what exactly these all peptides, proteins, which I am telling the biotechnology derived products, they are known as the macromolecules. Okay, so all your proteins, peptides, they are the natural molecules which we use for treatment of different diseases nowadays. The purpose of the drug discovery, we have to identify a drug candidate with unique profile to treat unmet needs of disease. Why we don't go to a lab and there are so many chemicals are available in your lab and use them, start using them as a drug? What will happen if you will start using them as a drug? And you don't know whether they are safe, whether they are what kind of toxicity levels they have. We don't know anything about it. So that's why we need to develop the drug molecules which are specific, which are having low side effects, and they will be able to treat that particular disease. If we can develop those kind of molecules, that is the drug molecule. So we need a drug which has defined pharmacology to address the unmet needs. So its pharmacology is defined, its mechanism of action is defined, its pharmacokinetics is defined, its absorption, distribution, metabolism, elimination, toxicity, everything is defined. Then only that particular chemical can become a drug. Balance drug-like properties to produce a therapeutic effect with an acceptable dosage regimen. So you will develop a formulation, you will develop a dosage form, but it should have a balanced drug-like properties. Adequate therapeutic window to side effects. Okay, so nobody will prefer the drugs, even your doctors will not recommend you a drug which has potential which has various side effects as compared to its benefits. Everybody will prescribe a drug which has high benefits, less side effects. That's the purpose. And limited pharmacokinetic and therapeutic effect variations in defined patient segments. Another question that is why we need to develop the drug is the drug should be novel, patentable. Okay, if a company is spending so much of money in developing a new drug molecule, you can imagine how much they will be earning. Okay, there are various drugs which have changed the company's lifeline. They have developed a molecule and that molecule was a blockbuster drug. Lipitor, which is one of the top selling, which was like in the, so if you go back to 2000s, it is like 2000, 2002, 2003 was one of the blockbuster drugs. Gleevec, which is used for the treatment of leukemia, ABL, which is the ABL kinase inhibitor. It is one of the blockbuster drugs. They have earned lot of money. Novartis, they have earned a lot of money with that drug. So that's why the companies, they want to develop new drug molecules that they will be having the exclusive right to sell that drug. They will be having a patent. They are able to sell that drug in the market for 20 years. And you can imagine in that 20 years, they can earn how much of that. That's the purpose. So this is key development process overall. First thing, you have to identify your target. Okay, target means you have to identify the receptor, you have to identify the enzyme, where your drug is going to act, where it will bind, what exactly it will do, what pharmacodynamics it will be having. That you have to understand. Then we identify the drug, we identify the lead molecule. We try to find out the lead molecules from where you will identify. We have a large number of chemicals are available. If you look into any of your chemicals catalog in your college, go to your chemical store, and if they have some chemical catalog, you see how many number of chemicals the company is producing. Okay, there is number of chemicals are available. If you will see one of the largest companies is producing the chemicals is Sigma Aldrich. Okay, Sigma Aldrich catalog, if you will see, it's such a huge book it is there, and maybe around millions of molecules there. So out of those millions, I have to find a hit compound or a lead compound, and this will be active against it. That is the lead identification. Once we identify the lead, we make the changes by synthetic chemistry, by using its molecular chemistry. We want to make that molecule to be more active. We want to make that molecule to have lesser side effects. It should have drug-like properties, everything. So that we do in the medicinal chemistry. And then we test with it in vitro. Directly, we cannot go to the animal testing and all. So we have to analyze it in vitro. We develop the biological assays in the lab. We develop the maybe the cell-based assays, or maybe the enzymatic assays in vitro studies, and we test our molecules against them. And then we go to the in vivo studies, and lastly the clinical trials. The clinical trials, all of you probably will be aware, they are basically different phases: phase 1, phase 2, phase 3, FDA review, and phase 4. So these were the news also during your Corona, when they launched, the different companies were launching the vaccines. There were clinical trials of, some of the clinical trials, we are not aware of the drugs which are being developed. India is not like, we are not developing so many small molecules and all, but vaccine was the utmost importance. We launched our own vaccine. India was also one of the countries which launched their vaccine very fastly, and they did their clinical trials also. Without those clinical trials, nobody will give the approval to launch the product into the market. We don't know the safety, we don't know the efficacy and all. So that's why we have to understand. That's why these clinical trials are planned. Phase 1, 20 to 80 participants, phase 2, 300 participants, phase 3, 1,000 to 3,000 participants. And once these are done, then we go to the regulatory bodies and ask them, okay, we have developed this molecule, we have come out with this biological product, I want to launch it to the market, this is the data, everything is there, it is safe, it can be used, it will treat only this particular disease, all those things we review, and then we launch that product. This is the steps which we use. So there are Now, as I told you, when you have to develop a new drug molecule, how you will go for it. Nowadays, nobody is like the times are gone when we found serendipity in drug discovery. Okay, we were knowing that something is like penicillin, it was a serendipitous drug. Okay, somebody was working on some microorganisms. Fleming, he was working on the microorganism, and he found that there are some microorganisms which are producing some chemical which is killing them itself. So he analyzed that. He found some chemical is being released by those microorganisms, and then penicillin came into picture. Okay, later on it was developed. Salicylic acid, urine, which we are using. That also is serendipity drug discovery. So many were there where we have like discovered the drugs by serendipity. But the times have gone now. We have understood our genetic system. We know these genes, how they are translating and how they are developing the different types of proteins. We know all the receptors. So why not to go for a rational drug design? Where we know already our target. Like this is the target and we want to develop a molecule which will act on this molecule. That's our control is. So we use these two different types of approaches. One is the target directed and another one is the phenotypic drug discovery. So target directed, where we use target is, you know, the target. We will validate that target, that this particular target is involved into this particular disease. So if you will see the cancer drugs which are available in the market and see their different mechanism of actions. So one is producing its mechanism of action by one route, another is doing another way, third one doing another way. Now which to use in which type of cancer? Okay, so that can only be done when we know exactly what is the target which is there in that cancer. So then we use that particular. We cannot use the same anti-cancer drug for the treatment of blood cancer. We cannot use the same anti-cancer drug for the treatment of breast cancer or any other type of cancer. We cannot use the same. We have to understand what exactly is there in that cancer, which gene has undergone the manipulation, the changes have taken place, protein has changed, all those things we have to understand. Then only we can develop that new drugs. So that is known as target-based drug discovery. Okay, and the second one is the phenotypic drug discovery. In this case, we know the disease. That disease has come. You have seen the COVID. Okay, in the beginning, when COVID came into picture, there was a hydroxychloroquine. Nobody was knowing whether it will act or not, but people started using it. Okay, nobody was knowing that where it will bind, how it will produce its mechanism of action, but people were using it. Okay, so that was phenotypic drug discovery, where disease was known, target of the virus was not known. Target of the virus came into picture later on. We came to know the different proteins of the virus very late, and then we started treating those viruses, those targets, and by them inhibiting that virus. So this is overall the drug discovery cycle. We start with the gene, we identify the target, we validate the target. Yes, this particular target, if we will inhibit, it will produce the mechanism of action. It will be helpful in that particular disease. That's exactly what we do. Then we go for the lead generation. We identify the different leads. We modify them, lead profiling, lead optimization, all those things we do. And then go for the preclinical testing, clinical trials, and then drug comes into the market. So these are the major steps in the drug discovery. First, target validation. Then we screen the compounds, secondary assays, and in vivo analysis. Now, suppose I'll give you a billion of compounds, and I will ask you to test them in the lab. So what do you think? How much time it will take? We're giving you billion of compounds. Let's say, for example, 100,000 compounds, and I'll ask you to test them in the lab. Go and check in the lab how many of them they are able to inhibit the microorganisms. We go into the lab and do some microbiological assays and then analyze how much time it will take. So maybe years. So can we reduce that time? Yes, we can reduce it by using the computational tools. That's why the computer came into picture. Can we develop some algorithms? Okay, can we develop what is an algorithm? Algorithm is it's a language using a computer language, you're developing a software. You are using a computer language, you are using Fortran language, C++ language, any other language which you can use, you use that, develop a software, develop an algorithm, and use that for screening of the compounds. That's exactly we want. So that will reduce your time. Those 10,000, 1 lakh compounds, 100,000 compounds, the time taken was years. I will be able to do analysis of those compounds in minimum 2 days. 1 lakh compounds, I can easily screen them by computational tools in hardly, you can imagine, like 2 days. And then out of those which are having the good potential, I can use them and test them in the lab, in the biological assay, in vitro settings. That's exactly we do. So we do the compound screening. Nowadays, the pharmaceutical companies, the big R&Ds, they are using robots also for test, doing this screening also. So the manual screening is avoided. They are using the robots. They will do the faster screening. So robot is unlimited, no time foundations. It will keep on working as long as it is. Till its electricity is on, so it will keep on working. So that's another way, that is known as hyperpush. Okay. And then we go for the in vitro assays, and then in the last in vivo analysis. So these are the drug discovery timelines. If you look at the different phases of drug discovery which I have mentioned, target validation 1.5 years, compound screening 1.5 years, lead optimization 1.5 years, preclinical testing phase 1, phase 2, phase 3, and approval to the launch, approximately 10 to 12 years is the time which a drug molecule takes to be launched into the market. So we have to reduce this time, and we have to reduce the cost involved also. The cost is also very high, $billions a pharmaceutical R&D has to spend in developing. So we have to reduce that cost also. So that can be done by using the computation. And the another problem is the success rate in developing a molecule, in developing a drug molecule is very, very less. Okay, hardly 5 or 10% chances are there that a drug has safety, efficacy, all those aspects, and then it will be we will be able to use for the treatment of a disease. So that's why the success rate is also less. So you have spent billions, and in the end, zero. Pharmaceutical company failed, all their money gone. You think why they will invest so much? So all those things we have to. So that's what I was explaining. Like the data is available nowadays. If you will go to one of the database, which is known as Chemical Abstract Services, which is maintained by this CAS registry. Okay, so American Chemical Society, they are maintaining this CAS registry. So we'll look at the number of chemicals they have in their database, in their library is 10 raised to power 16 is the number. I cannot calculate how much it will be. 10 raised to power 60 number of chemicals are known nowadays that can be explored for the development or for the discovery of any new drug good for a particular disease. This is the GenBank sequence. GenBank is a database of genes. Okay. So in this, we have the human genes, E. coli, and so many different bacteria whose genomic sequence has been resolved. They are deposited into this database. I gave you an example. Human genome, 3.2 million base pairs. So you can imagine what is the size of this. And then we have the availability of the proteins which are being produced by these genetic by the genes. So the number of proteins, as I told, is more than 100,000 in our body. They are available. We have explored the functions of some of these proteins, but as of now, many of them are unknown also. So many times, if you keep on reading the news and all, you will find that somebody has recently discovered a particular protein which is involved in this particular disease, and if you can use it for the treatment, that will lead to development of a new drug compound. So that's the PDB database where the number is increasing slowly. You can see presently it has more than 160,000 proteins whose roles have been defined, whose structures have been used, all they are available into this database, and we can use that. So we have the challenge now. We have a huge chemical space, which I mentioned, 10 raised to power 60 chemicals are there, which we can use them, like we can find out the hit compounds, we can find out the lead compounds, we can identify the lead compounds using this much of the chemical space. So if you will do this analysis in the lab, maybe our life will go, we will not be able to finish it. That's where the computational tools came into picture. And the targets, as I told you, 100,000 proteins are there in our body, which are functioning in different ways. They are producing different mechanisms, or in the form of different receptors, enzymes, your body building proteins, your structural proteins, and other things. So they are your biological targets. More than 100,000 targets. So out of those, you have to identify which target is required for treatment of a particular disease. And out of this chemical space, we require some molecules which can be used to target that particular protein. That is our purpose. We have to identify. So we have to choose the right molecule. Challenges I have defined. Okay. So these are the computational tools which can be used first in the target identification. One is the bioinformatics. So by bioinformatics, we know how those 25,000 genes, 25,000 genes, how they are coding different proteins. If we can analyze this particular gene is having this, but is responsible for the production or generation of this particular protein. That data is available. You can use pharmacophore modeling, protein structure prediction. You can build the proteins. Okay, so and then we can find out like whether that particular protein can be used as a target or not. In screening and hits to lead, we can use virtual screening, molecular docking, FEP free energy calculations, molecular dynamics, de novo drug design, lead optimizations. We use different types of QSAR. They can be 2D QSAR, 3D QSAR, 4D, 5D, so many are there. Pharmacophore modeling, molecular docking we can use. And in the last, where the development phase comes, we can use the different tools for the prediction of absorption, distribution, metabolism, elimination, and toxicity parameters of the compounds. And also we can do the PK modeling also. So we use this computer-aided drug design. One is if you know your target, that is one approach. So we can use the docking there. Another thing is if we do not know the ligands, suppose we don't know like which molecule is going to act, so we can use the de novo drug design approach. So we can use the receptor structure, protein structure, and we can develop the compounds based on the protein structure. So and then if the protein structure is not available, we have only the ligand structures available. In that case, we can use the ligand-based drug design, where we can use QSAR, pharmacophore modeling, and all those things. And if suppose both of them they are unknown, so then computer-aided drug design is of no use. Which I gave you the example also, like in the beginning of this COVID pandemic, nobody was knowing the structure of the virus, but hydroxychloroquine was used. Immediately, the people they started using it. So that was a random trial. Okay, so computer-aided drug design of no use, but we were using it. These are some of the informatics tools which can be used, structure-based docking, structure-based drug design. When the protein structure is available, we know the structure of the protein. So we use structure-based drug design, that is known as the we can use the approach algorithm that is docking. Another method is if the structure of the protein is not known, so we can use the pharmacophore-based modeling. So we have different compounds, we identify what are the chemical features in those, and we use them for the drug development. Then ligand-based QSAR similarity search, we can use that. And then knowledge-based chemical biology network, we can use the knowledge-based also. So this was the principle which was used in this had use of hydroxychloroquine. We were having some knowledge that maybe this may act, so why not to use it? Okay. So, one of the major steps when we start with the drug discovery nowadays is first step is the virtual screening, where we screen the library of compounds against our target preference by using different computational tools. So we can use some of the general approaches, like we can use, like I told you, benzene. But can we make benzene as a drug? Can it be used as a drug? What is there in benzene that it can be used as a drug? There is nothing. Okay, no group is there, nothing is there in benzene that it can be used as a drug. Suppose if I have a library of compounds, benzene and so many other chlorine and so many chemicals are there, I want to exclude them from there. So I will check their drug like so, whether it is having some drug like or not. So we can use drug like filters, which will eliminate those unwanted compounds, which are of having no drug like. So drug like filters, ADME filters, whether drug will be absorbed, distributed, metabolism is possible or not, elimination is there or not, toxicity it has or not. So all those things can be done. Then we can use ligand-based methods where we use similarity search, pharmacophore modeling, QSAR, and also your artificial intelligence, which in the uh, basic, it is called artificial intelligence, but it has some branches. One branch is known as the machine learning. So you are making your computers to understand. Okay, you are making your computer to work as a human brain. So that is machine learning. You are making your computer to understand. Okay, your Alexa, you are saying play this. What is that? Basically, it is play this song. It is analyzing your voice clearly, and it is a machine language, machine-based algorithm only, where we are converting our language into a process that is known as NLP, natural language processing. So language is processed into a work. So that's it. That's the output. Work is the output. It is playing the song, that is the output. But what is the input? Input is your voice. You are giving that as your input. So that is machine learning. And then structure-based methods, docking, diploma design, and free energy calculations. Some of the filters which you can use, as I told you, to eliminate the unwanted compounds. One of them is the Lipinski rule of 5, which can be used. So this rule says, so if suppose anybody is working tomorrow, maybe I don't know how many of you are doing the related to chemistry or not, but if anybody is working on developing some new compounds and all, you can use this approach. If you have a database of compounds, your guide is asking you to screen them, use this approach. Check whether those compounds are meeting these criteria. If they are meeting these criteria, then you proceed for the next step of computational analysis. So hydrogen bond donors, the number of hydrogen bond donors in a drug should be less than 5. Molecular weight of small molecules, it should be less than 500. Their log P or partition coefficient, it should be less than 5. Hydrogen bond acceptor should be less than 10. This is one of the filters which you can apply whenever you want to screen a large library. Okay. Then another method is, now suppose you have a already existing drug molecule is there in the market. Okay, ibuprofen is there, aspirin is there, or any other drug is there. I want to develop, my company where I'm working, they actually develop a drug molecule which is similar to, which is which will work like I do. So what you will do? So we can do one thing, we will use a similarity search approach. Two things we say, they will function if they are similar. They will have a similar function if they are similar, chemistry. If their chemical similarity is there, so they will function in a similar way. That's what is the chemical similarity. Can we measure the chemical similarity? How how we recognize one from another? Okay, by our characters. Okay, one has different face, one has small nose, one has big nose, one has big ears, one has this, one has that. Similarly, can we identify the similarity between your husband, your marriage is fixed, so we match the characters, 40 characters of this and this, they are matched, and if they are identical, then okay, both are married. So similarly, chemical similarity can, one chemical, if it is having the similarity with other, definitely, probably there are chances it will work in a similar way. It will have the similar reaction. If you will analyze all the different drugs, whatever you have studied, non-steroidal anti-inflammatory drugs, there are so many classes, but all of them they are producing the same mechanism. They are same effect, they are being used, they are inhibiting the same receptor. And if you will see the similarity between them, yes, there is some similarity. Somewhere they have the carboxylic group there, which they share with one. So we can measure the similarity. We can measure structural similarity. If we can measure the descriptor-based similarity, the molecular weight of two compounds is same, probably they are similar. If their number of hydrogen bond donors is same, actually they are similar. 3D similarity we can use, so you can generate the three-dimensional shape of a compound. If they are similar, it's 3D similar. Field-based, we can generate their molecular fields, electrostatic field, spheric fields, and see the similar. Two compounds, if they have similar spectra, yes, they will be similar. Okay. Similarly, quantum mechanics, if two compounds, they have similar energy, they have similar LUMO energy, HOMO energy, and all, so they are probably they are similar. So these are the approaches which you can use to analyze the similarity between two compounds. These all are the principles which are used in artificial intelligence, means developing the drug discovery tools as artificial intelligence based tool. These all are the principles which are used. That's why I'm explaining them. Otherwise, don't think what sir is teaching. Okay, so they are basically the tools which we use. We develop the algorithm, we develop the computer programs in a way that we will use them as the inputs, and they will analyze. Yes, there is a similarity between two compounds, and we will find some hits and we will use them as the final names for hit compound. So we can use structural similarity. It can be used in the substructure searching, maximum common substructure. You can divide the compound into fragments and see which fragments are common. These are some of the topological parameters which can be used. Okay, this B-cut is another type of molecular descriptor of parameter which can be used. So you can see the similarity between these three compounds, morphine, codeine, and heroin. All these three are very similar. If you will check, if you will put one on another, only difference will be in one, this OH is OCH3, in another that OCOCH3 and OCOCH3 is found. So otherwise they have the common framework. Okay, 4 rings and then one this pyridine ring, similar. But see the usage. One is morphine, which is narcotic, but very potential painkillers. Okay, one of the most potent molecule which is used as the painkiller, but it has a side effect also, that is the narcotic. Codeine, it has no painkiller effect. You have reduced the effect by changing only one group. You have changed this property. Antitussive. Heroin, no painkiller effect, completely none. Okay. So changing just the structure, similar structures, you have changed only one group, change the entire properties of that. That's the purpose that why we use the similarity searching approach. So all these things are there, which can be used molecular descriptors for measuring the similarity coefficient and all. This is the same way how we have the fingerprints. The computer we can develop the fingerprints by computers also for chemicals also. For each chemical, we can develop a unique fingerprint. Benzene has a unique fingerprint as compared to toluene. So that those fingerprints can be used to compare the similarity between two compounds. This is another example where you can use the shape similarity. So shape, here you can see this compound above one and the lower one, almost they are occupying the same volume. Okay, where they are very much sharing the same volume. That's why they have the same shape. Many of the drugs they bind with our receptors in a unique pattern, bind in a unique shape. Particularly, if you will look at the PPAR receptors, okay, where you have this roller lysozyme and those kind of drugs are there, which are being used for treatment of diabetes. They bind in a very unique pattern, that is a Y shape. So if any compound has a Y shape similarity, you can say that it is similar, it may have the similar mechanism of action. These are some of the examples of the similarity measure. So I will not go in, these are some of the work which we have done, so I am not going through all this. And the other method which we can use is the pharmacophore modeling. So one was the similarity search, another was the pharmacophore modeling. So if two compounds, they have the similar features. Okay, they have the similar number of hydrogen bond donors, similar number of hydrogen bond acceptance and all. So maybe their pharmacophore are common. So their pharmacophore are similar. So we can use one compound in place of another. That's what I was giving you the example of non-steroidal anti-inflammatory. Okay. Of course, it will, which I need a lot of time, but anyway, I'm trying to finish in 10 minutes. So pharmacophore modeling, this is one of the step approach which can be used. We have software available. They can be used to identify the pharmacophoric features among different compounds and then analyze which compounds they have the similar pharmacophore. We can use them for different receptors targeting. Okay, I'm not going to explain this. So another approach is the molecular docking. So as I told you, molecular docking is nothing. Okay, somebody has written a program which can understand what will happen when a drug is binding with the receptor. We have used a computer language, programming language, and that a algorithm is written, a software is developed that will understand what is happening when a ligand is binding with the receptor. Okay, when a ligand is binding with the receptor, its enzyme. So if we can understand those things, we can differentiate one ligand from another. All of you have studied enzyme substrate theory, where a particular type of substrate can only bind with an enzyme. So similar ways, particular type of ligand can bind with a receptor. If it has a different shape, it will not bind, it will not be accommodated there. So it will not produce this mechanism of action. These are the steps in the molecular docking. So generally we use large library of compounds. We try to bind them into the binding site of the receptor wherever your active site is. In that we will bind and we will analyze what kind of interactions are taking place between them. Okay, and then we can see which compound is better binding as compared to the other one, and we can whatever the best binding is, we can use them for in vitro screening. So this I am eliminating. So these are some of the AI applications. Okay, Netflix, everyone is aware of nowadays. Netflix, you will say, play this movie, play that, and after some time, once you have shown an interest that you are watching action movies, so next time whenever you will open the Netflix, it will automatically open for you that let's go to action movies because you have created login where you are watching those particular movies. So Netflix is what? It's artificial intelligence. Amazon Prime, another one. How Amazon, if you create your login into Amazon for shopping, okay, by using your email or Facebook account, next time whenever you will open Facebook, first thing will be reflecting there. You were interested in this product last time, now the prices have reduced. Okay, so maybe you are interested. So they have analyzed your mind, they have read your mind. The computer, which they have the system, they have read your mind that this person is interested in this thing. This person is interested in clothes, this person is interested in mobiles, this person is interested in this particular product on our website. So why don't we put this thing only there? Okay, HDFC Bank, Askiva. Okay, now all banks they have created their chatbots where they are also artificial intelligence based. So they have fixed answers. Okay, they will not give you the particular reason. Like if you will ask them why this thing has happened, they will not be able to answer. But some of the fixed answers already fixed, they will give you. Okay. Similarly, this car, which is artificial intelligence being developed, NASA they sent their spaceship stages. Okay, they all are artificial intelligence based products. Artificial intelligence in healthcare, very vast area, and it is being developed. Many of you are nowadays using your watches. They all are smart watches. They are also your AI-based products. Apple Watch, Samsung watches now they are able to develop it. They are able to interpret your ECG or how it is interpreting the ECG, from where it is getting the data. So they have a sensor which will read your movements of your blood. Then they will develop the signal. That's exactly what they are doing. So diagnosis, imaging, augmented reality, virtual networking, these all are the fields where artificial intelligence is used. Drug development, gene functioning, drug discovery, clinical trials, we are using artificial intelligence. Mining the medical data with artificial intelligence. Okay, wearables, the explained smart watches and all, they are all the things. Internet of medical things, which is coming up now. Another thing is it will have the maintenance of the patients, all the records it will be maintaining and tracking of the medications. Okay, so now this system has come. Okay, there is the different apps which are being NetMed and all. So they are using that data. Okay, every time they will ask you, last time you bought this product, maybe because you have a prescription which you have uploaded. So next time, after 10 days, you will get a message. Probably your medicine might be over. So you have to repurchase it. So why don't you buy it again? So they will ask you. So what is artificial intelligence basically? It's a process of using machine learning. As I told you, machine learning, which refers to system that can learn from the experience. Okay. So Alexa, as I told you, Amazon and all these, they are reading your mind. So they are experiencing. Somebody is feeding them. Okay. They are experiencing that this person is interested in this thing. That is machine learning. Deep learning. So if you have a very wide data, okay, the data is huge. You are using voice, you are using images, text, everything. So in that case, deep learning comes. Same thing, but the data is huge. Natural language processing, where we are developing where that can understand the language. So that is again your phone. Hey Siri. So this is the example. Okay, so we are using the techniques to build the artificial intelligence models that can perform high-level computations and solve the complex problems. So we have developed a system which is similar to which is like our brain, is functioning like our brain. It has the number of neurons which are there in our brain. It has similar number there also in the system you have developed using them. It will increase the accuracy. You can do high-level computation, you can solve the complex problems, and you can make the decisions. So many times nowadays, chess players, they are playing with a computer. So what is that? It is reading your mind. Many times, many of them, there is some like the good players, they are able to defeat the computer. Okay, but many times not. So that means he is reading your each and every move which you will make. So that is artificial intelligence. So we sense the world, we analyze and understand, we make the decisions. That is artificial intelligence. All these tools which I mentioned here, all these basic things, okay, similarity search, different types of filters, all of them, they will be your, what they will be? They will be your inputs in this case. So they are your senses. Your computer will analyze and understand them, and then it will make a decision. Okay, this molecule, I have this much of 10 raised to power 60 chemicals are there. Out of those, I want only 2 should be the most best one. So yes, you can give, but you have to give some input. What that input will be? That input will be some types of chemical features, some types of similarity, some types of filters, all those things you have to give as an input. So you collect the data, you store the data, process the data, and you get the informational insights, or you get the outputs. So that is known as the predictive model. So we have predicted. Your computer has predicted your behavior. Your computer has predicted your mind. That's exactly the artificial intelligence. These are some of the tools which can be used for artificial intelligence. Machine learning is, of course, it is there. And then you have under machine learning, we can use artificial neural network, decision tree algorithm, and other instance-based algorithm. These all are the different methods which can be used. This is the workflow for drug discovery, particularly I have included. So you screen the library using pre-screening tools, Lipinski rule of 5 and all. You do the similarity search, 2D or 3D similarity. You do the pharmacophore search. You do the docking. You do the hit identification, clustering, and evaluation. And then this is the lead identification. That's exactly this is it. How the drug discovery is being done nowadays. The time has changed कि भाई आपने पेड़ पे गए और एक बिगा प्लांट कर दिया है और यूज कर लिया. No. Nowadays, you are using medicinal plants. Okay, but have you heard like somebody has extracted the compound from there and then used that as a drug? Very rare. Only few compounds are there which are being used. Nobody is able to. Everybody is saying quercetin is very good. It has lots of properties. Nobody is able to use it. Why? Because it has issues. It has lots of issues, particularly with absorption and usage. So this is overall discovery flow. Anyway, you can contact Dr. Ajmer also for this all these things. These are lots of companies which are there, which are working nowadays in drug discovery with the artificial intelligence. They're using artificial intelligence. One of the recently company which came, that is AlphaFold, that it's a you give any protein as an input and it will develop this 3-dimensional structure basically. So this is one of them. These are others also. Several examples are there where artificial intelligence is being used. The artificial intelligence has really reduced the drug discovery time. One of the drugs which was like recently discovered was that with the help of this technique, in 1 year they have found a new drug. Of course, it's clinical trials and also will take time. But the other timelines they have reduced to 1 year, where in the normal cases it was 6 to 7 years. Okay, so thank you. Any questions, better? Anybody? You have seen now computer-based methods, how they are changing your things. I have explained drug discovery. Dr. Saleh, he explained you formulation development, how the statistics, how the computational theory can be used for reducing the number of formulations you have to develop. So the things are changing.
Okay, good afternoon, all of present here. I would like to thank Dr. Vijay sir for sharing the valuable knowledge related to the all of the artificial intelligence in drug discovery. Thank you, sir. And I think many of students will take the benefits from this lecture. Thank you, sir.
Thank you very much, sir. Now I would like to request Dr. Kumar Gaurav sir to please participate our speaker of the day.
I just want to mention one thing like Dr. Kumar Gaurav has taken the step to organize this conference.
Dr. Ajmer.
It's a very, very challenging task, to organize a conference nowadays. I really find like it's not easy, but they have taken this step, they have gone ahead and really, and you can imagine like the knowledge you people will be receiving will be of useful for your future career. You are saying?
How can we attach light on software?
No particular software, but what you can do is you can understand what the nanoparticle is, what is its composition. Then you can understand if you are attaching a ligand there, you can understand the chemistry between them. That can be done using the software. For example, like we have done one of the study where we analyze cyclodextrins that is used in the soil. How drug molecule penetrates and cyclodextrins. What will happen if we change that molecule? So how is the solubility will change? That thing you can do. That the way that is not possible.
I request Principal Sir to please also welcome our guest of honor of the day, Shri Dharmendra Singh Ji. He is a member Pharmacy Council of India, Jharkhand. Please, sir.
ये मैं गुड मॉर्निंग बोलता हूँ और मैडम को करेक्शन करता हूँ कि आई एम नॉट फ्रॉम ओनली झारखंड. I am the AC member of PCI and I am also representing Jharkhand as an executive member for the State Pharmacy Council. Please correct it.
I also request you, sir, to please say a few words.
मैंने तो तुम्हें नाम दिया. देखो मैं हिंदी अंग्रेजी में उतना ही तेज हूँ जितना कपिल शर्मा वो तेज है. ठीक है ना.
तो मेरा हिंदी बड़ा अच्छा निकलता है। अंग्रेजी बोलोगे तो फिर मुझे जाना होगा अंग्रेज के जमाने में। और भाषा वो होती है जो आप और मेरे को समझ में आ जाए, राइट है। बच्चे लोग जितने हैं थोड़ा सा एक बार रेज हैंड करना प्लीज, ऑल द स्टूडेंट्स, वेरी राइट। बिकॉज मैं आपको रिप्रेजेंट करता हूँ, टीचर्स को कम करता हूँ, आपको रिप्रेजेंट ज्यादा करता हूँ, ठीक है। आपकी जो भी क्वेरीज रहेगी आप मुझे जरूर मेरे मेल आईडी में लिखो, और जितना भी डंडा चलाना होगा कॉलेज को मैं चलाऊं, ठीक है। और एक बात है, जितने डेलीगेट्स आते हैं, तो मैं भी जब बच्चा था तो मैं बैक बेंचर था। सर अभी बहुत अच्छा एक अपना ये प्रोग्राम दे रहे थे और किसी ने क्वेश्चन नहीं किया, किसी ने, एक लड़की चंडीगढ़ की कोई लड़की थी, उन्होंने क्वेश्चन किया, वो हो सकता भूले भट के कुछ पूछ रहा था, पूछ दिया। अगर कोई प्रोग्राम होता है और आपकी क्वेरी नहीं होती है तो समझो या तो टैलेंटेड ज्यादा है और या तो सर कुछ समझा नहीं सकते। या तो आप लोग ज्यादा टैलेंटेड हैं, इस प्रोग्राम का आपका बेनिफिट नहीं ले पा रहे हैं, या तो आप इस ऑडिटोरियम के अंदर नहीं है, क्योंकि इतनी अच्छी प्रोग्राम चल रही थी, इतनी अच्छी स्टडी चल रही थी, मुझे नहीं लगता किसी बच्चे को ज्यादा मेरे जैसा समझ में आया। तो सर आई अप्लाई फॉर दिस वन फ्रॉम द स्टूडेंट साइड, क्योंकि इस क्षण को अगर आप मजा नहीं ले पाए तो आप किसी भी क्षण को मजा नहीं ले पाए। हमारे जमाने में ये सब कुछ नहीं होता था, और बड़ा मुश्किल से कभी कोई फैकल्टीज आते थे और कुछ लेक्चर्स देते थे, तो वो भी मुझे मौका नहीं मिलता था। पूछते थे कितना कितना ग्रेडिंग है तुम्हें, तो मैं बोलता था 60%, ठीक है तुम बाहर खड़े रहो 70 वाले को अंदर। एट दैट टाइम ऐसा होता था कि हम लोग बड़ी खोजते थे कहीं हमें ऐसे टीचर्स मिले, आप लोग जैसे अभिभावक मिले जो हम लोग को कुछ पढ़ाएंगे। और जिस जगह में आप लोग बैठे हो बच्चे लोग, बड़ा अच्छा हमें तो देखने में आया, बहुत सेशन चले हैं और आप बच्चे लोग कुछ ग्रैब नहीं कर पाए तो क्या होगा, आप सिर्फ क्या करेंगे, बाहर जाएंगे, सोशल मीडिया में अपनी फोटो डालोगे और लिखोगे हैशटैग पीसीआई, कुछ नहीं करना। पीसीआई को फिर दम भर के गाली दोगे, फार्मेसी कौंसिल इंडिया का दम भर के गाली दोगे, क्योंकि आपको पता ही नहीं है। आप गाली लिखोगे मुझे नौकरी नहीं दिला रहे हैं। अरे पीसीआई नौकरी दिलाने के लिए थोड़ी बैठा है, वो तो खुद नौकरी खोज रहा है, मैं भी नौकरी खोज रहा हूँ, मिलते ही नहीं है। चलो मेरे तहे दिल से आप सभी का धन्यवाद देता हूँ, क्योंकि आपके बहुत से टीचर्स आने वाले हैं, मैं आपके बीच में अवरोधक नहीं बनूंगा। और मैं आप सभी स्टूडेंट्स को आपको बताना चाहता हूँ कि आप लोग, आपकी आने वाली जनरेशन जितनी भी है, फार्मेसी काउंसिल इंडिया आपकी ध्यान दे रही है, और आप लोगों में एकदम आश्वस्त करना चाहता हूँ, जरा से इधर उधर किसी भी करिकुलम में हुआ, प्लीज डू राइट टू फोन मी, कॉल मी, टू योर प्रेसिडेंट और टू मी ऑल्सो। आपके ईआरसी चेयरमैन भी आए, मुझे नहीं मालूम आप लोगों ने क्या बेनिफिट उठाया उनसे, ठेकेदार सर से, क्योंकि आप लोगों ने कोई क्वेश्चन नहीं किया। उन्होंने, मैं जब पूछा उनसे कि सर कुछ आया वहां से, बोला तो आपके पास इनोवेटिव आइडिया, क्योंकि वो अकेला काम नहीं कर सकते। ईआरसी चेयरमैन जो है हमारे, अगर जब तक टीचर्स फैकल्टीज कुछ मदद नहीं करेंगे तो क्या फायदा होगा। हम तो आप लोगों के बीच में हैं, हम आपसे पूछते हैं कि हमें क्या सुधार होना चाहिए। आप बताओ, पीसीआई में भी थोड़ी है ना, हम लोग कस्टोडियन है, आपके बीच में हमें बैठा दिया गया है। आप लोग जो भी क्वेरी करते हैं वो हम लोग उसमें डालते हैं। हम लोग आपके इस सभा में आते हैं और सभा में इंटरेक्शंस करते हैं कि आपको क्या चाहिए, आपकी अच्छी सुझाव हम लोग पीसीआई में डालते हैं। अभी हमारे जो प्रेसिडेंट है मंटू पटेल सर, वो आपके बीच के ही है, वो भी स्टूडेंट जैसे ही करते हैं, उनको सीखने का बहुत आदत है, सिखाने का नहीं। हमारे ईआरसी जो चेयरमैन है वो भी सीखते हैं, हर एक बच्चे से सीखते हैं, वो जानने की कोशिश करते हैं, हर टीचर से मांगते हैं कि कुछ दो जो अगले वाला आने वाली पीढ़ी को हम दे सकते हैं। क्योंकि अभी आपको मालूम होगा, अगर ईआरसी सर बोले होंगे कि आने वाला जो कोर्स होगा वो होगा स्किल डेवलपमेंट पे हमारे फोकस है, राइट है ना। जैसे अगर आपको टीचिंग लाइन में जाना हो तो सेकंड ईयर के बाद आपका एक कोर्स उसमें, अगर इंडस्ट्री में जाना हो तो वो हम लोग उसमें पढ़ाने की कोशिश करेंगे। और जो कि हमारा गवर्नमेंट है वो भी यही चाहता है, स्किल डेवलपमेंट। उस पे जाना चाहिए। इसकी कमेंट्स आप सबों को आना चाहिए। आपका मेल आईडी है, बच्चे लोगों का पोर्टल मेल आईडी है, सर लोग जितने बैठे हैं उनको मैं इस पे रिक्वेस्ट करूंगा अपना अपना कोई भी जो भी आपकी सुझाव हैं वो हमारे पीसीआई में लो, क्योंकि देखिए सर हम भी उतने ही ब्रेन रखते हैं, आपसे कम ही ब्रेन रखते हैं जो हम चला सकें। और मैं अगर आप लोगों का कुछ भी सुझाव नहीं रख सका तो फिर मैं पीसीआई के ईसी कमेटी चाहे सीसी कमेटी के लायक नहीं हूँ। बगैर सपोर्ट का आप लोगों का नहीं चल सकता। मैं टीचर्स फैकल्टी से हाथ जोड़ के विनती करता हूँ कि सर आप लोग पीसीआई में एक अच्छा सुझाव भेजें और आपके सुझाव को हम लोग जरूर अपने आने वाले करिकुलम में उसको डालना चाहिए। इस रिक्वेस्ट और बच्चे लोग से एक रिक्वेस्ट है कि सर जब मतलब क्लासेस ले रहे थे जो आपकी चल रही थी उसमें से थोड़ा सा निचोड़ के कुछ ना कुछ पूछना चाहिए। उससे पूछने से दो चीज मालूम हो जाता है कि हम सोए नहीं है, पहली बात तो हम सोए नहीं है और हम कुछ ग्रैब कर रहे हैं। कितने दूर से सर आ रहे हैं, एमआईटी कॉलेज के एक नाम है, ब्रांड है, हर चीज है। वहां हम लोग भी जाने में डरते हैं दरवाजा खोलने में कि जाएंगे तो सर कुछ क्वेश्चन कर देंगे, हम लोग फिर बाहर भगाएंगे। तो ये सोचते हैं और आप लोग के बीच में सर आए हैं, इतना सरल भाषा में बोल रहे हैं, आपको समझ में आया या नहीं आया, मुझे भी नहीं आया, आपको तो मालूम नहीं आया कि आप लोग वन ऑफ द बेस्ट कॉलेज में पढ़ रहे हो। मैं भी अभी सर के साथ आपकी कॉलेज गया था, बड़ा अच्छा दिखा सर, बड़ा आई मस्ट थैंक्स टू द मैनेजमेंट ऑफ दिस कॉलेज वेयर आई वेंट टू दैट प्लेस। वहां से मुझे तुलसी का एक्सट्रैक्ट भी मिला, आई थैंक्स टू कुमार सर। लगता मुझे लगता है सर ने बोला कि आपकी ह्यूमिडिटी की कमी है इसीलिए आप ये तुलसी रखो। तो मुझे आई थैंक्स टू कुमार सर कि मेरे हेल्थ के लिए कॉन्शियस थे। और आपसे सभी बच्चों को बताता हूँ कि मैं ऑल द वे ट्रेवल करते रहता हूँ, कलकत्ता बंगाल में, कभी बंगाल से ही आया हूँ, पढ़ता हूँ, पढ़ाता हूँ, सबको सुनता हूँ और अपने जो प्रेसिडेंट है चाहे एसी मेंबर उसको भी डंडा चलाता हूँ। आपको शायद मालूम है कि नहीं, मैं अंदर से भी लड़ता हूँ, बाहर से भी, है ना। तो ये आप सबों के लिए, मैं बच्चे लोगों के लिए बोल रहा हूँ खास करके। आपको आने वाला जो सेशन होगा वो एक ही काम, एक ही आदमी मदद कर सकता। जानते हो कौन? जानते हो बच्चे लोग कौन कर सकता है आपकी मदद? आप खुद ही। और वो भी कैसे? ओनली स्टडी, ओनली स्टडी, ओनली स्टडी। अगर आप स्टडी से इधर उधर जाओगे, अभी तो डिप्लोमा में डंडा चला है, बी फॉर्म में भी डंडा चलाने जा रहा हूं। आपको मैं इसी मन से बोल के जा रहा हूं, मैंने डिप्लोमा में भी डंडा चलाया है, बी फॉर्म में भी चला। ये आने वाले आपको विद इन 6 मंथ्स में आपको मिल जाएगा। तैयार हो जाओ बच्चे पढ़ने के लिए। अगर आप नहीं पढ़ सकते हो आज ही नाम कटाओ, कहीं और चले। फार्मेसी को बदनाम मत, राइट है सभी बच्चे, राइट है ना। पढ़ाई करो, पढ़ाई करो, पढ़ाई करो। माँ बाप ने पैसा खर्चा करके आपको भेजा है यहाँ, कुछ ऐसा नाम करो कि हरियाणा का नाम बुलंद हो जाए, है ना। एक दो साइंटिस्ट में आप भी नाम आ जाओ। लास्ट में बंगाल में था एक पुरुलिया से सर साइंटिस्ट है जो कि कोविड के टाइम में उन्होंने बहुत अच्छा रिसर्च किया था, सीएमआर में संदीप नंदी सर। तो वो अपनी जब हिस्ट्री बताया था तो हम लोग के रोंग पे खड़े होंगे कि सर ये फार्मेसी का आदमी यहाँ तक आया है विदाउट फार्मेसी, इज नॉट ए फार्मेसी पर्सन। और वो हमारे लिए इतना रिस्पेक्ट करता है फार्मेसी के लिए, और हम अपनी मूल्यांकन भूल गए। ये तो बोलो भला बोलो कोविड का जो फार्मासिस्ट का नाम फिर से ला दिया, नहीं तो हम कहाँ पतन में थे मुझे नहीं मालूम। आपके यहाँ पत्रकार भी बैठे हुए हैं फर्मालू वाले, हमारे टाइम टू टाइम वो मदद करते रहते हैं कि फार्मेसी क्या है, फार्मेसी क्या है, फार्मेसी की भलाई कैसे होती है। आई मस्ट थैंक्स टू झाजी आल्सो फॉर दैट। फर्मा लोग के हैं वो। देखिए कुछ एक आदमी होते हैं जो हमारे इंडस्ट्री को आगे ले जाना चाहते हैं, इंस्टीट्यूट को आगे ले जाना चाहते हैं, तो उसको हर समय याद करना चाहिए कि भाई ये लोग हमें आगे ले जा रहे हैं तो उनको भी हमें सहता है। हमारे ना हम वो किसी एक पर्टिकुलर को तो नहीं ले जा रहे, एक फार्मेसी फ्रेटर्निटी को ले जा रहे, तो उसको याद करना चाहिए। जैसे सर ने बोला कि कुमार जी, गौरव जी और इस मैनेजमेंट को हर समय धन्यवाद देना चाहिए कि आज के दिन में एक कॉन्फ्रेंस करना बड़ा मुश्किल होता है। एक कलेजा होना चाहिए, एक मतलब पॉकेट भी होना चाहिए, तभी वो कॉन्फ्रेंस हो सकता है। और इतना खर्चा करने के बाद अगर बच्चे लोग कुछ नहीं ताली में जाते हैं और कुछ नहीं पूछते हैं, तो ये जो मास्टर होता है ना वो भी अपना पढ़ा के चले जाता है, घंटी का इंतजार करता है, जैसे घंटी बजी बंद किया चले गए। जब क्वेश्चन आंसर होता है ना तब मास्टर को भी मजा आता है, जो पढ़ाने वाले जो है ना उनको भी मजा आता है, यस इसमें से कुछ हमें बताने का, कल मैं और स्टडी कर है ना, क्योंकि वो जानता है कि मुझे कहाँ पढ़ाना है, किस लेवल में पढ़ाना। मास्टर्स जो होते हैं वो जानते हैं किस लेवल में पढ़ाना मुझे। आप आज 10 क्वेश्चन कर जाते ना भी, तो जो नेक्स्ट डेलीवरेशन देने वाले हैं ना वो तैयार हो गया, अरे यार ये तो बड़ी पूछते हैं, तो या तो मुझे स्किप करना है या तो भागना है, तो वो आपको आपके ऊपर डिपेंड है। और क्यों ये नहीं कि पूछना है कोई मुझे सर आपका नाम क्या है, ऐसा कोर्स में पूछना है, जो उन्होंने पढ़ाया उससे पूछना, ठीक है बच्चे। और अंततः एक चिट्ठी दिया गया मुझे, सबका नाम। देखिए मैं तो सबसे पहले मैनेजमेंट का धन्यवाद देता हूँ, और मैनेजमेंट बहुत ही अच्छा मैनेज कर रहे हैं, और गुरु गोविंद सिंह का नाम जो है वो हमारे बिहार से है, तो उनके लिए मैं सत श्रीकाल उनको भी बोलता हूँ। दूर है हमारे बिहार से, पटना साहिब से, उनका नाम को उन्होंने रखा है, तो मुझे मालूम है कि उनका नाम ऐसे ही व्यर्थ नहीं जाएगा, ठीक है। तो मैं मैनेजमेंट को तहे दिल, अगर मैनेजमेंट है तो उनको तहे दिल से मैं धन्यवाद दे रहा हूँ कि आप अपने नाम को बरकरार रखें, गुरु गोविंद साहब के नाम को। उनकी कॉलेज बहुत अच्छे चला, उनके मैनेजमेंट फैकल्टीज बहुत अच्छे, एंड आई मस्ट थैंक्स टू डॉक्टर विनय मिथार इफ इज देयर, प्लीज सर थैंक यू, एंड डॉक्टर रणदीप दहिया सर इज देयर, सर थैंक यू, डॉक्टर पूजा अरोरा थैंक यू मैम, डॉक्टर गजेंद्र नाथ तिवारी सीईओ फाउंडर्स जीएनटी पेज प्राइवेट लिमिटेड, थैंक यू सर। एक्चुअली आई मैं आपके साथ एक क्रॉस नहीं हुआ क्योंकि पेज मेकर लग रहे हैं, पेज तो मिस्टर सुरेश सैनी हेड एचआर इंस्टीट्यूट लिमिटेड इज देयर इंस्टीट्यूट कंपनी, थैंक यू सर फॉर प्रमोटिंग आवर, है ना। फार्मा इंडस्ट्री अगर हमें प्रोत्साहन दे रही है हमारी फ्रेटर्निटी को, उसको तहे दिल से मैं धन्यवाद देता हूँ। इंस्टीट्यूट कंपनी के मैनेजर्स को बोलेंगे, आपको भी मैं बोल रहा हूँ, थैंक यू। मिस्टर शत कुमार इंडिया एचआर मैनेज पीक्यूई, थैंक यू सर, थैंक यू फॉर प्रमोटिंग अस। डॉक्टर कुमार, ये तो अपना भाई है, कुमार गौरव जी हमारे छोटे भाई, प्लीज एक बार प्रोग्राम तालियां सर, सभी तालियां सुझाव कर ले। और मैं देखा हूँ, देखिए अभी कुछ एक दोस्त हमारे कुछ एक प्रिंसिपल्स बहुत सी जगह से आए हैं, ये सारे आ रहे हैं, इनके लिए भी जोरदार तालियां हो जाए। ये सारे ऑल द वे आपके लिए, कुछ भोपाल कुछ ग्वालियर, ये सारे आपको ये लोग भी ऑर्गेनाइज करते हैं, बट आज का जो कुमार गौरव जी का प्रोग्राम है ये मैं इसी मेंबर और ऐसे हमारे अभी आए नहीं है ईआरसी चेयरमैन, बहुत ही मजा ले रहे हैं और ज्ञानवर्धक है, इसको मजा लेंगे बच्चे लोग। और जाते जाते एक ही चीज बोलता हूँ बच्चे लोगों के लिए, इवन द टीचर्स फैकल्टी आल्सो आई रिक्वेस्ट, आप दोनों भाई से वर्षा है, अगर आप टीचर्स पढ़ाना चाहेंगे तो बच्चे पढ़ेंगे, अगर टीचर्स नहीं पढ़ाना चाहेंगे तो आजकल बच्चे इतने एजुकेटेड खुद पढ़ लेते हैं, राइट है ना बेटा, मतलब खुद पढ़ लेते हैं, या आप क्वेश्चन मार्क मार रहे हो कॉलेज के ऊपर, राइट है। और भी अगर आपको कोई क्वेरी चाहिए, पीछे से कोई मदद चाहिए, अभी भी पूछ सकते हैं, हम आपके लिए मतलब अवरोधक का काम करते हैं। अगर आपको कोई भी क्वेरीज हो, है ना, अगर पीसीआई से रिलेटेड, पीसीआई के कोई फंक्शनिंग से रिलेटेड, ये मत पूछना मैं पीसीआई गया तो चाय नहीं पिलाया, क्योंकि फार्मा लोग को पहले भी आदमी जाते हैं, सरकार लोग को उनको भी चाय नहीं पिलाते हैं, सुनते आजकल, पहले चाय पिलाते थे ये लोग, भी नहीं पिलाते। बच्चे लोगों को क्वेरी हो तो बताओ जल्दी, और नहीं तो मैं यही पे प्लान करूँ अपनी बातों को, क्योंकि आप लोग बड़े टैलेंटेड आदमी, आप लोगों के पास कोई क्वेश्चन नहीं रहता है क्या बच्चे, नहीं है ना। एक चीज बताओ, कोई कोने से लाल हाथ तो उठाया, मैं बताऊं पीसीआई का मतलब जानते हो, वो है पेस्ट कंट्रोल ऑपरेटिव, पेस्टिसाइड बनाते हैं ना, क्या क्या करता है फार्मेसी काउंसिल ऑफ इंडिया, एक बच्चे बताओ, बच्ची को बताओ, आप बताओ, आप सर मैडम दोनों बीच में, इधर उधर देख लो, आपसे मैं पूछ रहा हूँ, हाँ फार्मेसी काउंसिल ऑफ इंडिया क्या करता है, घर में भी ऐसी बोल, जोर से बोलते हैं। अच्छा अभी गला करे हो, चलो थैंक यू, आप कुछ जानकार हो। और कोई बच्चे बैक बेंच में? आप तो कुछ क्वेश्चन कर रहे हो ना, मैं पूछता हूँ। वो पाजी किधर गया, पाजी किधर गया? नहीं आप नहीं। ओ पाजी उधर उधर। सर जी मुंडी क्यों छुपा रहे हो? बताओ बताओ, कुछ जानते हो फार्मेसी काउंसिल ऑफ इंडिया के? बताओ, फार्मेसी स्टूडेंट हो कि टीचर हो? स्टूडेंट हो, अच्छा बताओ फार्मेसी काउंसिल ऑफ इंडिया क्या करता है? रूल्स बनाता है, दाएं जाओगे कि बाएं जाओगे? वेरी गुड, बढ़िया बेटा। देखो क्या होता ना कि मैं इसलिए पूछ रहा हूँ क्योंकि मैं जब यहाँ हरियाणा नहीं सॉरी यूपी गया था, तो यूपी में बहुत हमसे ज्यादा टैलेंटेड आदमी और हर आदमी नेता है। तो हमने पूछा सर पीसीआई क्या था? वो कुछ नहीं सर, कुछ नहीं होता है, अप्रूवल देता है। तब तो भाई हम बोले ठीक है, क्योंकि वहां बोलना भी बड़ा मुश्किल है, क्योंकि कब बाहर में सर आपके लिए यूपी में ही आता है। वहां बोलना भी बड़ा मुश्किल है कि बाहर में गनमैन खड़े रहते हैं, निकलना भी है, घर भी जाना है, हमारी भी रिस्पॉन्सिबिलिटी, मेरी भी शादी हुई है ना? तो मुझे भी सेफली घर जाना। हम ठीक बोल रहे हैं, अच्छा आप ठीक बोल रहे हो, फार्मेसी काउंसिल वाली अप्रूवल देता है और इंस्पेक्शन करता है। और मैं जब टीचर से पूछता हूँ कुमार सर, तो एक ही क्वेश्चन करते हैं कि सर मेरा इंस्पेक्शन कब लगवाओ? नहीं नहीं, मैं सर से पूछ रहा हूँ क्योंकि सर के घर पे हूँ, तो पूछ लेता हूँ कि क्वेश्चन करना चाहिए कि नहीं करना चाहिए, क्योंकि आगे भी तो जानना है। तो वो जितने टीचर्स है ना सारे ही मेरे नाम जानते हैं, सारे मेल आईडी फोन नंबर जानते हैं, कहाँ से निकालते मुझे नहीं मालूम। जब भी मैं रात को सोने जाता हूँ तो 2-3 फोन कॉल आते, सर मैं यहाँ से हूँ और मेरा 14 ईयर्स का एक्सपीरियंस है और पहले लगता था अब नहीं लगता। तो हम बोले क्या लगता था? बोले इंस्पेक्टर। तो हम बोले पहले देते थे अब नहीं देते, इसीलिए नहीं लगता है। तो ऐसा है कि जिसका भी इंस्पेक्शन है, जो भी टीचर्स हैं जो क्वालिफाइड हैं, हमारे मंडू सर का एक रिक्वेस्ट है आप सबसे, मजाक अपनी पार्ट पे, कि आप लोग हमारे मेल आईडी में अपनी एक्सपीरियंस के साथ भेज दें और सारे को अप्रूव किया जाएगा और सारे को इंस्पेक्शन मिलेगा, चाहे एक मिले चाहे 3 मिले, मिलेगा जरूर। और सब इंस्पेक्टर से आप जो भी एक हमारा क्राइटेरिया दिया हुआ है उस क्राइटेरिया को फुलफिल करते हैं तो उसको सब मिलेगा। 10 चाहोगे 10 मिलेगा, लेकिन लेना क्यों चाहते हो ये बता दो। आपके जैसे टीचर्स, सर आप मतलब बताओ 5 लेने के बाद 5 लेना चाहते हैं, 10 लेना चाहते हैं, क्यों? घर नहीं बनी है क्या? सिर्फ घर नहीं बनी है क्या? अच्छा मैं भी मजाक ही कर रहा हूँ। चलो कोई बात नहीं, थैंक यू ऑल द वे। आप लोग ने बड़ी प्यार से जो एक क्वेश्चन सर एक बच्चे का क्वेश्चन था हम तक कुछ लोग बता। अच्छा कंप्लीट भी कर दो भैया, जब सर पढ़ा देते तो कंप्लीट कर दिया नहीं। बोलो बोलो बोलो। ये तो मेनका गांधी से पूछ। हाँ आपका पढ़ आपकी क्वेश्चन कल ईआरसी सर आए थे, ईआरसी चेयरमैन आए थे, आपने उनसे नहीं पूछा था कि पूछने नहीं दिया था उन्होंने? हार्ड आदमी है लगता है। वो आपकी बात मैं सुन लिया और आपकी बात भी सही है। देखो बेटा कहीं तो टाइम पास की जगह चाहिए ना, कहीं चाहिए। तो वो फार्मेसी काउंसिल ऑफ इंडिया ने बड़ा सोच समझ के रखा है कि आपको कैंटीन नहीं जाने दिया जाता, आपको कुछ नहीं जाने दिया जाता। एक टाइम पास की जगह चाहिए जहां आप बैठ के फेसबुक में डालोगे और हमें पूरे गाली देना, ठीक है? लेकिन आपकी बात वैलिडिटी, मैं कुमार सर से बोलूंगा इस क्वेश्चन को फॉरवर्ड कर। और भी क्वेश्चन ये खोद खोद के निकाल रहे हो कि मतलब खुंदस निकाल रहे हो? चलो पूछ रहे वेरी वेरी थैंक यू। देखिए क्वेश्चन वो होता है जो सॉल्यूशन के साथ है, तो आई मस्ट थैंक्स टू यू। व्हाट्स योर गुड नेम? थैंक यू। आई आस्क मैं रिक्वेस्ट करूंगा कुमार सर को कि सर आज मेल कर दें, दीपेंद्र सर से मैं बात करके आपकी बात को मैं करवा दूँ, ठीक है? और कोई क्वेरी हो तो बताओ। कैंटीन में खाना मिलता है कि नहीं बताओ। चलो थैंक यू। वंदे मातरम, भारत माता की जय। और ठीक है, थैंक यू, जय हिंद।
Thank you very much, sir. Sir, your presentation given. Now I request Dr. Kumar Gora sir, Pushpendra Singh Naruka sir, and Karni Singh Shekhawat sir to please come up and facilitate, sir. नेक्स्ट लेक्चर वैसे एनुअल स्टडीज के ऊपर ये है, कोई भी क्वेश्चन हो आप नेक्स्ट सेशन में पूछ सकते हैं। Now we shall be meeting after 15 minutes for the next lecture. There will be the tea break. After tea break, we shall be meeting there again.
आज साफ ये बैठे थे, साफ